Hello, 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 everybody. I was muted. Welcome
Wait, I
to the
think I'm
stream.
muted too. There we go.
No, I think you're all good and clear. I actually unmuted you before I unmuted myself. Welcome, everybody. Welcome to the stream. Hello. We have a very special guest today in the Octopus Lady, who has been so generous to give us some of her time so that we can talk about a topic that we both mutually have a deep love for, and that is the pufferfishes Tetrodotoxin, a very potent neurotoxin that I cannot wait to talk more about. But before that, please, special guest, why don't you introduce yourself?
Hi, everyone. I'm the Octopus Lady, and I am a YouTuber who makes videos about marine biology and all the weird and wonderful things that live in the ocean. And one core aspect of my videos is everything I talk about I needs to be backed up by peer-reviewed scientific research. So I'm really excited to be here with Phi today.
Yeah, I'm excited to be here with Octopus Lady today, especially because, you know, my audience knows this about me, but I am a pretty fiercely competitive person by nature, and I have a little secret to share. Some of your videos make me go, oh, God damn it. It's so good. It's so good.
Yeah. Thank you. That's so sweet of you to say. And I totally empathize. Like, yeah, there's been times where I've watched your videos and I'm like, that was really cute and clever.
Yeah,
Like,
it's just like the knuckle biting of like, damn, that is so charismatic.
yeah.
But
no,
yeah,
there's been times where I've been watching your videos and I've just been like, wow, he explained that really well. That jerk.
Yeah, exactly. I think anybody who makes content, this is like the quiet part that they don't say out loud, but I actually think
Yeah.
it is more honest to say that kind of thing out loud, because it's like, I think it's like a very high form of respect when I watch one of your videos and I have to go, damn it.
Yeah. Yes, I totally empathize. I totally get where you're coming from.
So for those of you who are from my audience who might not be so familiar with your work and who you are, I figured it would be good to start stream just by asking you a couple of basic questions about you and your content.
Yeah.
I think what was really amazing about talking to you behind the scenes was the fact that we both have the word biologist in our name, but we actually come at biology from very different angles. And I kind of wanted to explore what it was like, and maybe I'll contrast your educational upbringing with my own, because I find that like the assumptions that I make about other biologists are often very unfounded. So I'll just kind of start off by asking you, so what got you into marine biology? Like, when did you get into it, and what was sort of the deciding factor?
Yeah, so I don't know if this is a very exciting story or very anticlimactic, but had no interest in marine biology at all throughout most of my life until I was about 16 years old. I was just channel surfing, which for maybe the younger members of our audience, that's I was just watching TV and just like flipping through random channels, and this random documentary popped up on screen about octopuses. And for some reason, I watched it, and by the time the documentary was over, I was like, I need to know every single thing about this animal, and that was kind of it. I just knew at that point, like, I need to dedicate my life to this. And what's actually really funny is that when I was very, very little, like maybe like 2 or 3 years old, my parents would take me to our local aquarium, and they had a giant Pacific octopus tank, which was this huge, like dark tank that had a giant animal in it. And my parents could not get me near that tank. As soon as they would start walking towards it, I'd immediately burst into tears. I still viscerally remember the fear of being near that tank. So when I told my parents, oh my God, you guys, I think I know what I want to do for the rest of my life. I think I want to be a marine biologist, and I think I want to study octopuses. My parents were like, what? Um, so yeah, I don't know. It was one documentary. That was all it really took. It was kind of wild.
Do you remember which documentary? Kind of a
No, I don't. And I'm so sad because yeah, that was like, whatever it was life changing. It was on PBS. I do remember that. So, shout out PBS.
I think it's really interesting beyond the fact that you essentially conquered the fear and made it your entire discipline, we were actually talking behind the scenes a little bit about the idea of kind of the macabre, kind of dark aspects of biology being an inroad to studying it very sincerely.
Yeah.
And like, there's nothing more terrifying to me than the ocean.
Yeah, I mean, honestly, on some level, I am still deeply terrified of the ocean, but I have also discovered about myself that when I am terrified of something, I also weirdly need to learn every single thing about it. Like, I could tell you, it sound random, I could tell you so much about the video game Silent Hill 2 because I was foolish and watched my brother play that game at 3 a.m. when we were teenagers and proceeded to have nightmares about it for years. But like, I could tell you all about its development, its production, the plot, the symbolism of the plot, because there's something about terrifying things that my brain is just like, maybe if we know enough about it, it won't be scary anymore. That hasn't really worked for most of the things I'm scared of, but it actually kind of works for marine biology. Like, I'm still scared of the ocean, but not as scared as I used to be, if that makes any sense.
It does, and we're actually gonna be talking about that phenomenon of humans exposing themselves to unpleasant stimuli for the sake of just being human, right? Because I mean, I can relate to that in particular. If anyone's watched my rabies video, I did not grow up religious, but I did end up in a Korean math camp that was also like a church, and so my very first experience with religion was very secondhand by, summer school high schoolers who were basically just telling me that, like, oh, if you pray, then you will have your wish granted. Essentially, I thought God was like a genie. And every night I would pray that I would not get like a prion disease or rabies. And
God,
I just have a laundry list.
do.
But that's the same kind of feeling of like, this thing terrifies me, and it's very human to want to make that your whole thing.
Yes, absolutely.
That's awesome. So this is like from one biologist to another. It's sort of, I don't know if the audience finds this kind of interesting, but like, did you feel like you had to delay your passion because marine biology is not offered in a real way until later in your education? Like, did you have like a feeling of impatience essentially before actually studying marine biology?
That's an interesting question. No, I don't remember feeling impatient. I'm trying to remember because pretty, I was decently into my third year of high school, or secondary school, I think is what the Europeans call it. So I was only like a year and a half away from going off to university, and there, the internet was just to date myself a little bit. The internet, was starting to become a thing in households when I first got interested in this. So I was able to kind of satiate my own interest in marine biology for like a year and a half, before I went off to college.
Did you so when you were in college and studying marine biology, did you like have so here's so I often say this about medicine to give you some context, is the idea that like being a doctor is like so different than studying biology. You don't really get the chance to try on the clothing of a doctor unless you somehow manage to land like a hospital internship. Then you can sample what that life was like. Do you feel like, did you have like research experience in undergraduate, or did you have opportunities to try out marine biology before deciding that was what you wanted to study?
No, nope, just no. I just the second, yeah, I just as soon as I was introduced to octopuses, I was like, I have to make this my whole life. I have to make this my whole personality. I did do a couple internships in college, but they weren't as marine biology focused as I thought they were going to be. They were a lot. One of the I'd say the most significant one I did was more phylogenetics than anything, which is really not my scene. So yeah, I didn't really do any hands-on marine biology research kind of stuff until my senior year of college, where we had to do like a research project.
Well, that's actually really incredible in terms of the fact that like you had this intuition. Okay, because the reason why I like exploring this kind of thing is because I have made it like my part-time hobby to talk pre-med students out of being doctors. It's just kind of been like a hobby of mine to be like, do you really know what it's like being a doctor? But you're actually in a situation where something about the octopus resonated with you, and that has remained. Like, it's kind of like that idea of when you show up to a cat, like a shelter, like the cat picks you, like you just know which cat is right for you. There's no checklist. And you would say that this connection was very sincere, pure, and didn't require a lot of reflection or thinking.
Yes, correct. It's very, I will say this in one aspect of my life where I'm very lucky. I do not seem to agonize over really, really, really big decisions. So like what I want to do with my life, or like the second, like my car, the second I saw my car in the used parking lot that I was going to eventually buy it from, I was like, that's my car. Like, I'm going to buy that car. Or like when I saw my apartment that I currently live in, after seeing dozens and dozens of other apartments, I was just like, this is my apartment. Like, but what's also very funny is that like I can go to IKEA and like look at 8 different tables that are like $100 each and be like, I don't know which one to buy, and then I just go home. Like, there's something about in general about me where like really, really big, huge decisions, I seem fortunate enough that I just always inherently seem to know. which
That's
one is the best for me.
that's
Not to brag or
so
anything.
that is so like enviable in a kind of way, because what this is communicating to me is that you have like a pretty good sense of intuition, not intuition for everything like life period, but you have a very good sense of self intuition. Like you know yourself, and you don't need to because like here you go. Like I just looked at a bunch of apartments and then I just picked one that felt right. I was like, I was also recently looking at apartments. There's a whole spreadsheet and data and compare and contrast and a matrix, a payoff matrix and everything. Like,
Yeah.
I get way in my head about these kind of decisions, but that's actually really cool.
Yeah.
And it's also refreshing because I live in this space of like I talk pre-meds out of becoming doctors. That sometimes you just actually just know. For some people, that just works. That's crazy.
And also, like I was saying, like I've literally spent 6 hours in an IKEA, couldn't decide on anything, and then just left.
I mean, that
Like
means
so,
it does mean that none of those pieces of furniture resonated with you. They didn't pass the vibe check, right? Like that's valuable in itself, right?
I guess not, but I really needed a table, man.
But you needed your table is the thing. You weren't gonna settle for second place. That's awesome. That's really funny. So I wanted to ask, speaking about like life path and stuff, that what was it that made you want to sort of do YouTube in this capacity?
Yeah, so, again, to date myself a little bit, I was on YouTube basically when it first, like, I was watching YouTube basically when it first appeared on the internet, but like for several years, the YouTube was just like the funny viral video website or the funny cat video website. It wasn't until like 2011-ish, I think, when I started getting into the Vlogbrothers. Um, and I was like, "Oh, like, YouTube isn't just a place to watch funny videos, it's like a place where I can be educated and also" we didn't call it this back then, but build parasocial relationships with people, for all the pros and cons that come with that. And, through the Vlogbrothers, I started watching a bunch of other YouTubers like, CGP Grey. Who else was I watching back then? Oh, man. I'm not gonna say, I don't wanna date myself anymore, and just so for years and years and years, I was just watching all my favorite YouTubers and just being like, I wanted, I wanted to do what they did, basically. And when I would try to go find videos about marine biology, I would basically, like, find 3 different kinds. Like, one kind would just be, like, it wasn't marine biology, it would just be people, top 7 things that scientists don't want you to know are at the bottom of the Marianas Trench. Like, number 1, Atlantis. And I'm like, this is nonsense. The other types of videos I would find would be videos very much targeted towards children, which is important. Like, it would, like, I think it's important for little kids to learn marine biology, but like, I do feel like ever since Finding Nemo came out, marine biology has kind of gotten this reputation of it's a science for children. When it's not, it's very much not. Like, yeah, there's horrifying things in the ocean that you shouldn't tell your children about because they will get nightmares. So like that was fine, but like the third kind of marine biology channel I would find, and I'm sorry if any of those channels are watching this stream right now, but like their videos would be static images with just text on screen, and just stock music playing, or they'd be static images of someone speaking, but they just had the most monotonous voice and they would just talk like this for the whole entire video, and they would be saying the coolest stuff like, pufferfish have toxin that can kill people in less than 30 seconds. And I'm like, how could you say that so boringly? Like, put some life into this, man. So what actually eventually ended up happening, sorry, this is a long story, but at my job that I had before I was a YouTuber, I was actually a trainer. It was my job to train new people in my company in what I did, which, by the way, had nothing to do with marine biology. I'm not going to say what I did, but I was working in a tech company. And it was my job to train the new kids who would come in. And we we would have this thing, this we have our all hands meeting for our department. And where like everyone in the department would have to come into a room and we'd have to talk to each other for an hour, right? But we would do this thing where for the first 3 minutes of every all hands meeting, you could sign up to do a presentation, and it had to be 3 minutes long, but you can present on anything that you wanted. It didn't matter what. It just present on something that you can educate your fellow coworkers about. So I made a 3-minute long presentation about sea stars, and I presented it to my department, and everyone thought it was the funniest thing in the world. And I actually remember like the head of my department. So I presented my presentation, right? Everyone was very impressed, not to brag or anything. But the head of my department, who was due to speak after me to like basically start the meeting, like literally said out loud, like, I don't want to go now. I don't want to follow that up. Like that was I don't know what to say. That was so good. And I'm just like, oh geez, thank you. But I took that 3-minute long presentation and then I just expanded it out into a 7-minute long YouTube video, because like it was a PowerPoint presentation, right? And like, as you know, Fi, like we make videos that are basically just really fancy PowerPoint presentations. So, you know, I made it into a YouTube video. I showed it to some friends and some family, and they were like, this is really good. You should post it. And then I did. And then basically what happened at that same job, I kind of I really liked that job for a long time, and then I didn't. And my performance started to drop as a result of me not liking that job anymore. And again, in one of those moments where I just knew immediately what to do, basically my boss said to me, like, you can either stay and try to improve your work performance, or you can leave and we'll give you a severance package. And I was like, well, if I stay and I don't improve my work performance, will I still get the severance package? And they were like, no, we'll just fire you. And I'm like, okay. And literally, I was like, for about 2 hours, I was like, oh my God, what am I going to do? What am I going to do? I don't know what to do. I don't know what to do. And then like, I don't know, at the 2-hour mark, I was like, I know exactly what I'm going to do. I'm going to quit my job and try to become a YouTuber. And here we are.
Yeah, I mean, it's an insane story.
Okay, I'm glad it's interesting.
It's an insane story, but it's also like very inspiring, especially because I was put in a very similar position, although I did not have the option to stay, right? But it is nice because we did talk behind the scenes, and I, you know, spoiler alert for chat, I already heard this story, but it is what I was alluding to, because I've referred to this story, or I've referred to this interaction before on stream, but like part of why I felt more comfortable trying to do YouTube is because of support from other creators like the Octopus Lady. So I would like to thank you for that. And also reflecting on what you were identifying as a relative lack in the marine biology space on YouTube, I think that is something that I've also been thinking about actually as recently as yesterday, as I forget who it was in my Discord, but thank you for the kind comment of, you know, I recently released a video about the long-term E. coli evolution experiment, and the comment was essentially saying that it was a very good example of the modern YouTube explainer style, which I found to be interesting because I never really thought about what that meant. And I think that hearing you say what old, you know, quote unquote old YouTube lacked, I think it's helped me to come to a sort of conceptualization of what that modern style actually is. And I think you're a very good representative of what I'm about to say, which is, it sounds like when you in your videos, it sounds like you are sharing something very special to you. And I think that is not common, even with some larger channels that I won't name. It is niche, but in a very passionate kind of way, because the way that I think about my content, I had a very similar experience as you in the sense where molecular biology content was mostly studying for the MCAT.
Yeah.
I think what's so nice about the modern YouTube explainer is that you know, in the landscape of YouTube of competition, and there's a finite number of eyes and you have to compete for those eyes. If you
Yeah.
are doing this kind of modern style where you are making videos in a way where it showcases your love for the subject and it sounds like you're sharing something nice, that I think makes the pot bigger, because you're not all competing for the same MCAT students. You are putting yourself out there, and I think the videos reflect that kind of loveliness, and I think that's very special.
Thank you. I've actually gotten it's interesting because I've actually gotten some comments from people saying that I remind them of old school YouTube, kind of for that reason, because that's what was really on YouTube at the start, was just people talking about stuff that they loved.
Oh no,
And
don't do
then
this to me. I refer to it as old YouTube, but that's really medium old YouTube.
fair,
No.
fair. um just like before monetization, that was what YouTube was like a place for people to talk about what they loved, and I still think it is. Like I don't want to be one of those people who's just like, oh, like, social media has gotten so terrible. It's not great, like there's a lot of problems with it, but I still think there's still a lot of value in it, and there's still a lot to enjoy. You just kind of got to go digging for it a little bit more than maybe you had to in the past.
Yeah, algorithms have made the most appealing thing the most popular thing, as opposed to the most personal special thing, right?
Yes.
By definition, because this work is special, it might not appeal to every individual who is scrolling through the algorithm, but it will appeal to a lot of people who identify kind of with that kind of content. I think that's very, very it's very nice. And yeah, I agree. Like, in terms of the algorithm, it is not the hellscape I think that people make it out to be. It's just different than it
Yeah.
used to be.
Yes. I agree.
Yeah, Morju says it's our kind of special. Dang it. Yeah, exactly. Like, that I think is, I've been thinking about ecosystems a lot, and not only for the collab, but just in general for YouTube. And I think that is more of a way to describe the modern algorithm. It is trying to build ecosystems rather than mass appeal, and I think that's interesting.
Yes.
With that being said, why don't we go ahead and talk about some pufferfish?
Yes.
Does that sound
I'm
good to
always
you?
down to talk about Yes, I'm always down to talk about pufferfish.
Awesome. So let me go ahead and set up. So I will actually begin the slides because the first couple slides are mine. So just letting you know that. And let's transition to this space where I will pull up my slides. Hello, slides. Oh, that's not the correct one. Is it this one? There we go. All right. So I had this slide up because I just wanted to spotlight again the fact that when we first talked to each other to try to plan a collab, usually those talks last for not that long, like half an hour. We brainstorm ideas, set up a date, schedule some stuff, and get a vibe check. But we actually talked for like 2 hours for the first time that we made contact, and I think part of why that conversation is so meaningful to me is because you are perhaps the furthest biologist from me that I have ever had contact with.
Yeah. Same.
Before that, it was probably Nep, where Nepenthes VT, who's a part of Ethereum. She's a horticulturalist, so working with plants. But like, I've worked with plants before in my training and my education. I have only vaguely learned about one or two fish, for example.
Yeah.
And to me, that like difference is that like we both call ourselves biologists, but what we practically do is very different. And I find that to be fascinating, and it has been a really interesting topic of conversation.
Yeah, yeah, definitely. Like, it's interesting because obviously you're a biologist. It's very clear, but like to me, what you do is almost more like chemistry. And I'm sure that there are chemists and molecular biologists who might be deeply insulted by that, but to me, it's just so different from what I do that what you do almost looks like, yeah, like more like chemistry than biology.
Which is actually a pretty fun segue into my next slide, which is
Yes.
this thing that I made. So don't take this too seriously. But when I was talking to the octopus lady and getting a sense for like what marine biology actually is, like I don't, okay, oceans, cool, but I had a lot of questions about like what that actually looked like in terms of what is actually done or what problems are actually addressed, which was very eye-opening for me. And part of what struck me is like you can very easily tell if you look at the pinned comment. That's the octopus lady's video on Tetrodotoxin. She gets into like some pretty hefty molecular biology chemistry in that video, actually further than I would have expected. There was actually a part of that chemistry that I did not know before, and it's the part about TTX having a certain charge in a certain location on its chemical structure, which is what allows it to plug up the hole. So I actually did not know that before. So I was and this is like what gave us the idea to do this collab to begin with, because this is sort of the perfect overlap between both of our interests. But what surprised me is that the octopus lady probably knows a lot more about molecular biology than I do about the ocean.
I'm not going to argue that. I will say, if there are any aspiring marine biologists in the chat, one of the best things I ever did when I was in college was not just take marine biology classes, like taking physics and chemistry and organic chemistry and molecular biology classes. I think all really helped contribute to making me a better scientist as a whole.
And I think that's sort of like what we got out of our initial conversation too, was that like, so sorry to continue the interview, but I do think that this is an interesting perspective that some up and coming scientists in the chat might want to consider, is sort of the scope of what you like to practice, right? Because for me, I would prefer to hone a couple of different tools and use them all over the place whenever I can. And it sounds, you can correct me if I'm wrong, but it sounds like in your education for marine biology, you actually have to accumulate way more tools than I say that I would have to, for example.
Yeah, we kind of talked about this briefly, and I guess so. The thing is that like I don't really look at what I learned from biology as like tools I can use. They're just really I don't know. It's just cool stuff. So like yeah, like yeah, because like you were talking about how like what's cool about molecular biology is that you can learn a handful of things and then you're kind of good to go. Um, and yeah, that's definitely not the case with marine biology, but that's what I like about marine biology is that like you're always constantly learning new things. If that makes if I'm if I was understanding you correctly.
Yeah. So to kind of expand on that kind of idea, is it's that like when looking at something, I mean, I could have a myopic view of my own discipline too, but when I look at molecular biology, I can chunk them pretty neatly into mostly these 5 categories of subdisciplines, and then there's overlap and subspecializations, etc. But like when I read a cell biology paper, I can classify it as cell biology. I can remember the tools that I need to interpret a cell biology paper, which overlaps with genetics, but like I kind of view it as like, oh, to read this paper, I need to pull out this toolkit, et cetera, et cetera. But I think from what I gather about marine biology is that there are so many, if you were to read a marine biology paper, you might not know which tools you need to understand the paper immediately. It might take you to, might take you down all sorts of different avenues that you were not expecting.
Yes, that's accurate. Unless it's like unless I know exactly what I'm kind of getting into, like when I was doing research for my hydrothermal vents video, and I was trying to figure out how the microbes at the vents work, I would go into these papers knowing like, okay, let's dig up that really old like cellular biology knowledge from my freshman year of college. But overall, it's just usually when I like pick up a paper, I'm like, we'll see. Like, maybe this will like, I remember when I was also doing research into mantis shrimp and how their limbs work. I just remember being like, oh, this will probably be more like an anatomy paper, like when they're talking about their limbs. And then I would start reading it and be like, oh no, this is an engineering paper. This is not anatomy at all. I mean, there would be aspects of anatomy, but like, yeah, you don't. For me, that's like, I don't know what I'm getting into whenever I pick up a paper.
And I think that's an interesting contrast, because I would say that my research process has generally been pretty straightforward. The hardest video I had to make was on influenza, but that's not because I had to learn something new, just because influenza is such a confusing virus to begin with. But I didn't have to like acquire a new skill set to learn about that. I just had to deepen my understanding of virology. Whereas it sounds like when you do research for a video, it's like, oh, I guess I got to wear the chemistry hat today, like or engineering hat today. It sounds to someone who is not afraid of the deep waters, I guess, of academia, to put it metaphorically. That sounds appealing to a certain type of person, but I like knowing that I'm going to be able to get like 80% of a paper upon first read.
Yeah, like it's definitely like that would be great, but like there's also the yes. So that I wish I had that. I wish I had that. I wish that was more of a thing in my experience in marine biology. Because it can be really frustrating sometimes to read a paper, and I know this paper is really important to my video, but it can literally take me like 4+ hours sometimes to like figure a paper out. And I don't, and there have been times where I've spent 4+ hours on a paper and realized, oh, this paper actually doesn't answer the question that I wanted to answer. So like I wish I could just like I wish I was able to do that. That would make my life a lot easier. But the flip side to that is that the number of times I've been like surprised by what I found in a paper, which man, that high of like discovering, quote unquote, discovering something that, as near as I can tell, no one else has talked about on the internet. And like, yeah, it's the best. But yeah, it's like there's definitely positives and negatives to it.
I relate to that feeling so much. The feeling of like, did anybody talk? And like the fear of searching the YouTube video. I had that for the prion video where I was like, I'm sure people have made prion videos before, obviously. Like, it's one of the most scary diseases. And when I was looking through like the videos that were published already on this topic, they were of the medium old YouTube style, like pretty sterile,
right?
You know, not that deep, like very basic. And they were very popular, right? More views than what my video got. But like, I was like, no one's done like a deep dive on the prion thing, and I could feel the algorithm begging for it. Like, it is so,
right? Yeah.
So exciting. Okay. Yeah, that's funny to hear from another creator. It's very, very interesting. Okay, so I will move on to that next slide. So here's the title sequence. So the question that we sort of settled on was this one: why doesn't the pufferfish simply succumb to its own poison? So the pufferfish, if you had watched the required reading for this stream, does not synthesize its own TTX. It did not evolve to make it and store it in a way that is safe. It takes up TTX. I'm spoiling a little bit in the future, but just so that the problem is well communicated, it takes up TTX exogenously, is the fancy word from the outside, and so it is exposed to TTX the same way that we are, in a sense, where it is an outside thing that gets inside. But the difference between us and a pufferfish is that we are dead. Like, and this was kind of an interesting question, and when we were planning out what to do, the little digging that I had seen was that this is still very much a question that is not fully answered. I kind of teased this in a stream a couple of weeks ago where I was like, oh, people are not 100% sure all the details about how this happens. Whereas to me, I was, when I read this, I was like, surely people know this already. Like, surely.
Nope.
But in reality, it's actually not as clear. I mean, I think people are getting very close, but I think the full picture is still quite hazy. And in terms of the rigor that a scientist needs to say something is true, we will actually be encountering the limitations of what we know. And I think that's very, very exciting. And as a bonus sort of part of this presentation is that when we get to the segment where we hypothesize together about why, what the answer to this question is, we're going to do a little bit of a choose your own adventure, although there are only two branching paths. But I will now switch this over to the octopus lady slides so that we don't have to do the next slide thing that collaborative presentation mandates. Octopus lady, if you could full screen yours, I see that is already done. Fantastic. So give me 1 second and I will share that. Okay, so it looks like your slides are live. No issues there. Take it away.
All right, so first off, let's talk about what is a pufferfish. And it is not what is currently being shown on screen. This is a porcupine fish, which are related to pufferfish. Many people think these fish are the same, but they're not. Including Phi, who told me that he just grabbed the first stock image he saw of a, quote, pufferfish and slapped it on these slides.
Hugely embarrassing.
But pufferfish and porcupine fish are actually from two different families. Pufferfish are from the family Tetrodotoxin, while porcupine fish are from the family Diodontidae. So let's just take a minute just to point and laugh at Phi for getting this wrong.
I would just like to address a comment that I saw fly by in chat. I did watch her video is the thing, but the thing is we talked like literally like 6 months ago. We talked some ridiculous number. Like to plan this collab, we did it like half a year ago, and that's when I watched the pufferfish video. So I did apologize in private Discord messages, being like, I should have rewatched the video before I sent those slides, because you say that in your video.
Yeah. It's okay, man. You're a molecular biologist, so
yeah, we're a little slow when it comes to full organisms.
I'm just saying, is that like only cool people know that like pufferfish and porcupine fish are different from each other, and like molecular biologists are kind of inherently uncool. Like, no offense or anything. Like, it's just
on my own channel.
Yeah. Fans of Phi, I swear I did not agree to do a live stream with him just to humiliate him in front of an audience. I'm just poking fun. And in fact, it was all Phi's idea to have me clown on him a little bit for thinking a porcupine fish was a pufferfish. So if you're upset with me for teasing him, you should in fact be upset with Phi, because again, this was all his idea. So please don't be mad at me, because I get really sad when people are mad at me.
It was my idea. To be clear, she was very nice. She was very kind about the mistake. I just thought it was so funny. I was like, dude, I just started these slides. I just shared them. And immediately she was like, I was like, oh,
you know what,
honestly?
Honestly, man, when you sent me the slides, there was some gut part of me that was like, I bet he used a porcupine fish. Because people always, like, it was kind of really annoying to get some of these stock images that you're seeing on screen right now, because when I typed in pufferfish into, like, the stock image websites that I use, the whole first page was porcupine fish. Like, on almost every single website I went to. So you're not alone.
Yeah.
If that makes you feel any better.
Let's blame the search engines. Let's go.
Okay, so pufferfish are so named because they puff. They suck in water or air through their mouth, and that inflates their stomach, which is very elastic. And fun fact, they don't inflate like this. Like, they don't inflate around their spine. But like this. This is the X-ray of a porcupine fish that's inflated with both water and air. The water is the opaque part. The air is the transparent part. So their stomach just expands and expands and expands to where the fish can't even keep their spine straight anymore, which looks unbelievably uncomfortable. Pufferfish puff as a defense mechanism. Not many things can swallow you when you're the shape of a balloon, although some birds have been known to pop pufferfish with their beaks before eating them. Their other defense mechanism is their toxin, their Tetrodotoxin.
Yay!
Tetrodotoxin, which is abbreviated as TTX, is actually named after the family that pufferfish come from, Tetroodontidae, because researchers initially thought that tetroodontids, or pufferfish, were the only animals in the world that have TTX. But TTX has been found in a bunch of other animals, including the porcupine fish. Some of them have tetrodotoxin too. Newts, flatworms, the eggs of horseshoe crabs, sea stars, and finally, the best TTX possessing animal in the world, the blue-ringed octopus.
Can we get some octopi in the chat?
Hey!
Let's go.
Octopus gang represent. Squid Squad represent.
Oh, Squid Squad, that's good.
Yeah, there's actually like a small movement I've seen of squid researchers trying to push that when you see a group of squid, we should call them a squad, because right now they're called like a shoal, which is lame.
Oh, that's boring.
Yeah, Squid Squad represent. So what happens if you're poisoned by TTX? So Phi is going to talk more. It's going to go into more detail about what happens on a molecular level if you consume tetrodotoxin. But on a macro scale, TTX poisoning basically causes you to become completely paralyzed. And interestingly, people assume this means that all your muscles go rigid, but in fact, the opposite happens. All your muscles go slack. And I mean all of them, including your diaphragm. Treatment for TTX poisoning is extremely limited. If you can get your stomach pumped very quickly, and I mean very quickly after ingesting TTX, you might be okay. Otherwise, oh boy, strap in, folks. It's gonna be a rough day. There is no antivenom, so your only option is to wait for the poison to work its way out of your system. Fortunately, that usually only takes about 6 to 24 hours, but since you're paralyzed, you do have to spend those 6 to 24 hours hooked up to various life support machines so you don't die. What's even more fun is that even though your body shuts down, your mind apparently does not, as patients have reported being completely awake and aware of everything that was happening to them after becoming paralyzed by tetrodotoxin. So if you do end up poisoned by TTX, I guess that gives you a good opportunity for you to try practicing meditation while you are a prisoner trapped inside of your own body.
You literally have nothing better to do.
Yeah. Despite this, some humans looked at a pufferfish and were like, wow, a tiny amount of this thing's poison can kill me. I'm gonna eat it. Because humans are gonna human, I guess. In Japan, pufferfish are considered to be a delicacy, where it's called fugu. It is illegal to serve fugu there unless you are a sushi chef with at least 10 years of experience and have a special license. TTX is highly concentrated in different organs in pufferfish, which Phi is going to mention later. But there are trace amounts of it in their muscles, which get sliced very thin and are served raw. Apparently, eating fugu makes your lips tingle and also kind of makes you feel a little drunk. And speaking of being under the influence, you may have heard that dolphins get high off of pufferfish toxin. And I'm sorry to be a buzzkill, but I have found absolutely zero evidence to back this up. While dolphins have been seen playing with inflated pufferfish, it is next to impossible to determine the intentions of an animal that we cannot communicate with. Dolphins might be passing a puffer around because they're taking turns getting high, or they may be passing a puffer around because it's shaped like a ball, and a lot of animals enjoy the simple pastime of tossing a ball around. Now, I'm not saying... oh, excuse me. Now, I'm not saying that dolphins don't get high off of pufferfish. I'm saying that we don't know. So maybe we shouldn't go around saying that they do as if it's a proven fact. And then finally, tetrodotoxin may have ties to the origins of zombie stories, but if you want to learn more, you should go check out my video about pufferfish, which is linked down in the description. And with that, I'm going to pass it back to Phi.
Thank you so much. I actually find that the part of your video where you talk about the zombie stuff, I've wanted to do that for things like tuberculosis, but I also found it to be very difficult to do that kind of research. 'Cause I'm not going to spoil the video, but you do go into a bit of a rabbit hole.
Yeah. Like, I do go into a bit of a rabbit hole. I will also try not to spoil it. But it does involve studying humans and culture. And I don't like studying humans. We're way too complicated. Like, there's so much debate. Like, a group of humans does a thing, and there's so much debate about what that group of humans actually did, because people within the group say they did this, people outside the group say they did that. Two different people within the same group say different things, and it's like, but inside, but like, when like in the type of research that I do, it's like brittle starks do this weird thing. Why do they do this weird thing? It's because of X, and then everyone goes like, that makes sense, and then we all just move on. Like, humans be doing too much. Like, we got like yeah, it's really hard is all I'm gonna say.
Yeah, and that's why I actually chose not to talk about that in my tuberculosis video, because I actually find that a very valiant part of the video where you do the deep dive into the cultural aspect of TTX, because I'm not brave enough to do that. That seems like a lot of work.
Yeah. It was really, really hard. My pufferfish video is probably one of the hardest videos I've made, along with my most recent video, which is about gooey duck smuggling, because that was basically a video that was all about humans. And humans need to calm down. Everything I've learned about humans is we just need to calm down.
I'm very, very true. So true. All right, so let's get into the stream topic, which is, okay, so we know that pufferfish have this toxin. Why don't they succumb to it? And so let's go a little bit into the molecular details of this story. So Tetrodotoxin is an extremely potent neurotoxin. We already talked about this, that the pufferfish stores in its liver. And as the octopus lady actually helps me understand, it's not just the liver, it's also the skin, the gonads, and the intestines. So I did a little digging and I found the oral median lethal dose, or the LD50. So if you are an aspiring pharmacologist or doctor, this is an extremely important metric by which you can measure the lethality of a certain substance. This is the required dose to kill half of a specific population. This is the LD50 for mice. So I do say this every time when talking about animal research. It is not to be taken lightly. We don't do this for no reason, right? And it is very important that we in the lab make sure that we do this responsibly, safely, and with as little pain or misery as possible to the treatment of lab animals. I always make a point to point that out, just so that, like, for both sides of the spectrum, right? For people who are worried about animal welfare, in the US, the animal welfare laws are quite strong with regards to lab animals. And then for people who don't think about this kind of thing, I just want to remind them that every time you see an LD50, that is the result of animal experiments. So just when we work with animals, we should be as kind as we possibly can in the name of scientific advancements.
Well said.
Thank you. For a 70 kilogram man, or 154 pounds for us Americans, this is the equivalent of 21 micrograms of TTX to kill half of a given sample of full-bodied men, right? And 21 micrograms is such a small amount of powder that you can barely, like, I tried to find an image of what 21 micrograms of salt looks like, and you would need a microscope for that kind of thing. So it is very, very small amount. So Octopus Lady teased this in her post, but I actually worked with this toxin during graduate school, and my advisor used to constantly remind me that we have enough neurotoxin in our fridge to kill our entire town. And I didn't believe her at the time. I thought that was maybe hyperbole, but doing the math for this collab, I was like, oh, we had way more. Knowing the number of people that lived in our town, we had way more. And so it is very important to be able to, and like, I didn't tell you this before, but like a little bit of a story from when I was working with TTX is the first day that I worked with it, I was so scared. I was like, and there was nobody in the lab to teach me because they had all graduated. So I just had to read the safety protocols and double check with my professor to be like, hey, I'm going to walk you through exactly what I am going to be doing with this chemical, and you just tell me if I'm going to kill myself.
Oh my God. That's so terrifying. Oh my God. I could never. Like, I would have to watch someone do it for a while before I was comfortable with it.
It's not any different than any other dangerous chemical. It doesn't aerosolize, so that's good. It is you treat it as if it were a deadly liquid, which is different than a deadly liquid that can aerosolize, which then you need more safety equipment. I wore way too much safety equipment. I was like stacked with lab coats, and I considered double gloving, but you actually shouldn't for reasons, right? They can shear, the friction can cause them to tear, and like even though I had visual confirmation on this poison the entire time, I know I didn't get any of it on me. I had a lab coat, gloves pulled down past the sleeve, etc., etc. I left that lab, and I'm a very psychosomatic cell. So just knowing that I worked with TTX, I started to feel as if I were poisoned by TTX. I'm like, do I feel weak right now? Like, it's so scary.
Are all my muscles going slack? Like,
Yeah,
am I gonna be
yeah.
a prisoner inside my own body? Like, oh my god, I could. You are a braver cell than I ever would be.
It was. I got used to it over time because I had to use this quite often, but the first time was so scary. Yeah, 20 times more deadly than cyanide. So definitely not. It's a miracle that an organism, a complex animal evolved resistance to this. So just making the stakes clear.
Can I ask if do you know, like, if you got it on your skin, would that be enough to poison you, or do you actually need to consume it? Like,
So
do you need to put it in your mouth?
So that's a great question. I don't know the answer to it, but while I was doing research for this collab, I actually found applications for TTX as a topical anesthetic, as in they're trying to manufacture a formulation containing TTX to treat pain, like in a cream form. So, I mean, kind of extreme. Got to make sure that they dose it exactly correct, right? But I think that if I had gotten it on me, it probably would have felt like an anesthetic, although I don't know that for a fact.
Right. Yeah. Okay. I was curious.
Yeah, it is scary stuff. Okay, so why evolve TTX resistance to begin with? I think in the non-marine biology world, I think when we hear about TTX, it's usually in the context of the pufferfish and not usually associated with those other organisms to, the casual viewer. And so it's kind of an interesting question to ask, where does TTX come from to begin with? And why would evolution hone in on its ability to resist poisoning? So at first, the most obvious, I think, is chemical defense, right? But to me, okay, so the papers that I have read have said that this is obvious, and I think I don't understand animals well enough to really get a deep understanding of this, because, like, when a pufferfish, I don't know if you know the answer to this. This is a question I just kind of had on the spot, but like, if an animal were to attack a pufferfish, would they come into enough contact with it to act as a deterrent, or do they have to eat the puffer to actually suffer? Because you could imagine that, like, a lot of fish aren't super well. Okay, let me before I spread fish misinformation, my assumption is that fish are not that smart, and that pufferfish don't become that they don't get a reputation, right? That fish that encounter a pufferfish don't tell their fish friends, hey, don't mess with the pufferfish, that it is a defense. So I don't know if you have any thoughts on that.
Yeah. So so wait, sorry, can you repeat the original question?
Yeah, so
my mind went into like 10,000 different directions. So,
no worries. So as a chemical defense, I'm wondering if it's actually that effective because of the way that you have to ingest the TTX. Like, do they slough off enough of it to act as a deterrent, or is it something else?
That's a good question. I don't know if I can speculate on that. I don't know if I know if we know the right answer. Like, I know that a lot of people assume a lot of scientists assume it's a defense mechanism because stuff like toxin is almost universally a defense mechanism in animals. Like, if an animal has a toxin, it's for defense. But I'm assuming that there is enough poison in them that even just attacking them is enough of a deterrent, because they have a lot of it in their skin. I do think that you do have to consume it, though, in order for it to affect you. I don't think they need to eat the whole animal in order for it to potentially off a predator. I'm assuming.
Yeah, given how little you need it to actually kill, that does make a lot of sense.
Yeah. Yeah.
Uh, the second reason to evolve TTX resistance is access to what I call forbidden foods. So TTX accumulates in the pufferfish due to their diet. So you've actually you go over this in your video, where if you give a pufferfish a diet consisting of things that do not contain TTX, then they are TTX less. So we have pretty good evidence to show that in the wild that they are accumulating TTX in their environment, and it's not diet, or that it is diet dependent. So eating smaller organisms that bioaccumulate TTX, starting from, and this is something that I learned in your video actually, starting from the bacteria Vibrio, but we'll actually go over how strong that evidence actually is from a molecular biologist perspective, which I think is kind of interesting. So yeah, we'll save that for the end of stream, because I think that I think I make a lot of assumptions as to where different scientists from different disciplines draw what counts as good enough evidence. For molecular biology, the standard is like really, really high for what we adopt into canon. And I don't know if that's the case for other disciplines, but I'd be curious to get your read on if we seem like tryhards.
No, I have a lot of thoughts about that, but we could talk about that later in the stream.
Yeah, I think that'd be a great like end of stream discussion after, because like this is an example of us like sampling each other's discipline through a shared topic, and I'd be curious to hear what you have to say about hearing from me, even though you are more familiar with mole bio than I am. But this is like graduate level mole bio stuff that we're about to cover.
Gotcha.
So there's some evidence out there. This was really surprising, that TTX might be used as a pheromone. I say I hedge it so hard again because the standard for proof has to be very high. But there's some evidence from the 90s that hasn't really been expanded upon, from what I could tell, that pufferfish can use TTX as like a chemoattractant. Like you release it like from a source, and pufferfish are actually drawn to the source of the TTX if you dissolve some of it in the water.
That's really cool.
Whether or not it is true, I do not know. I make no claims on that, but there is evidence that suggests that could be a evolutionary adaptation to it.
Yeah.
And then so in order to sort of understand how this resistance happens, we first have to understand the molecular mechanism. So if you are here from the Octopus Ladies channel, hi, I should have done my intro way earlier, an hour earlier actually. But hello, I'm Fi the Neutrophil. I'm a cell molecular biologist. If the Octopus Lady is working with actual animals, I'm looking at the cells that make up those animals and the molecules that live inside those cells. So we live at different scales, essentially. What you're seeing on screen, this is the Octopus Lady, but I'm under a microscope that you can't see. And so to dig at why the pufferfish doesn't die immediately due to this, we have to look at the actual molecules. On this slide, you can actually see the molecule of TTX here. Okay. Oh, Extra Gimified has a comment that says, if they are attracted to the smell of TTX because it reminds them of food. That's an interesting hypothesis. I think it's in the stream description. I think I listed my sources there. I don't remember if I put that particular source there, but I think their argument was for mating behavior, not necessarily food. Yeah.
Interesting.
Okay, so how does TTX work? So this is an animation of a neuron that you can find on Wikipedia, and this is what is called an action potential. So an action potential is an electric signal that propagates down the body of a neuron. So it starts from the head of the neuron, goes to its tail, and this electrical current is, I think you explain it much better in your video than I'm about to do here, but essentially the way that you conduct electricity is you make sure that there's a propagating wave where charge is changing, right? So over the body of the neuron, you have sodium channels, which allow for the intake of positively charged sodium ions, and then there's a relative charge on the outside. So on the inside of the neuron, it is relatively negative. There is a relative difference where on the inside it is more negative than the outside, and when those sodium channels actually open, you get sodium in. Or is it sodium out? Dang, I should have looked this up. This is so bad for me.
I do know this because I barely explain how action potentials work in my video because I didn't really get them, but I do know this. So what happens is that sodium channels open, and then sodium comes in, and then stuff happens, I think, and then potassium channels open and a bunch of potassium gets let out. I don't know if that helps at all.
No, you're good.
Okay.
I was a neurobiologist, by the way. I just haven't thought about the action potential in a very long time. Yes, that is correct. You jogged my memory. The sodium channel opens. That causes positively charged sodium ions from the outside to flood the neuron, and then you can see that in that local area, you have a relatively positive charge. And in doing so, those positive charges actually causes the potassium channel to open, which then lets out potassium, and you get a relative evening out of those electrical charges. So this happens locally within the neuron, and this signal propagates down that body, and when that signal reaches the end of that neuron, that can trigger some electrochemical reactions that will release neurotransmitters to propagate that information to the next neurons. So the action potential is basically what you have to get out of this slide is the action potential relies on electricity, and that electricity relies on the opening of the sodium channel, this protein that allows sodium into the neuron. And so TTX, the mechanism by which TTX works, is on this sodium channel. So what we are seeing here is two views of the sodium channel that you can find in mammals. I think this is from a human, but I don't really remember. This left thing, let me have a draw function. I keep on forgetting about that. So on the left here, we have the top-down view. So into the screen facing you, like towards you, that is the top of the protein, and then out of the neuron is, I think I had that reversed. Into the screen is into the neuron, and out of the neuron is facing you. And here we have the hole that the sodium ion can flow through. And so if we take a side view of this, we have out and in. The sodium ion starts on the outside, and then it enters the neuron through the sodium channel. And so TTX causes neurons to shut down because it actually plugs that hole. So so Tetrodotoxin plugs that sodium hole. So you can actually see that in this picture right here. That's TTX, and it so perfectly sits in that hole and prevents that signal from being propagated.
I remember first learning about TTX, actually, I think in a molecular biology class I took in college. And I remember my professor saying that like tetrodotoxin acts as a plug, and it like plugs the sodium ion channel. And I'm like, okay, I'm pretty sure he's just being metaphorical there. And then it wasn't until several years later where I was doing research for my pufferfish video, where I was reading a paper about how TTX works with sodium ion channels. And like that actual paper was just like, it literally acts as a plug. That's literally what it's doing. And I was like, oh, wow, it's that simple? Because my assumption is always just like when things happen on a molecular level, it's super complicated. But no, it's just like, yeah, it's just like, plugging up your drain or something when you want to take a bath. I was shocked at the simplicity of it, I guess is what I'm trying to say.
I think that's sort of like the whole gist of my entire channel.
Yeah.
Is that like molecular biology, when you say it like that, it sounds very complicated, but in every video, I make sure to say molecular biology is just jiggling shapes bumping into one another. And this is another one of those examples. Look, all, this deadly poison, you might think it's all complex chemistry. In reality, it's got a shape and, it fits in the hole, and that's why you die. Like, that's really it. It's not that complicated.
Yeah.
So there's a question in chat: how many channels need to be blocked for the signal not to be transmitted? So there is a minimum voltage change that needs to happen to trigger an action potential, and as long as the charge remains static, like as long as you don't cross that, I don't remember what that threshold is, but there's a threshold of electric charge, and if enough sodium channels are blocked, you can't get to that charge where you trigger the action potential. Let me go on to the next slide. So remember that accidental consumption of this poison through pufferfish can lead to disastrous consequences. So when you plug those sodium holes in the neurons, those neurons will no longer respond to signals, and they will no longer propagate those signals to other neurons. So essentially they become deafened and silenced, right? This is kind of existentially horrifying.
Yeah.
I often find myself, like, as a molecular biologist, really, I don't know if you do this, it'd be kind of interesting to compare notes, but when I hear about stuff like this, I like feel for the neuron itself. Like there's something visceral about, you're talking happily to your neighbors, and then you're just you know, gag and blindfold and earplugs and everything. Like there's something visceral about it. And when I read molecular biology, I often find myself sympathizing, like with an infected cell that's been infected with a virus. I don't know if you do the same.
Yeah, no, I mean, what a miserable way they're a cell. They don't have a brain. I don't even know if they can experience pain. But the thought of like getting taken over by a virus is like the movie Alien, right? It's like that levels of horrifying. Like I totally get where you're coming from. Like I remember I experienced something really similar when I was doing research on frogfish because they can swallow something within seven milliseconds, like which is for reference, the human blink, like the time it takes for a human to blink is 100 milliseconds. So like to be a victim of a frogfish, you're literally just chilling, like you're a fish chilling somewhere, and then literally like in less than the blink of an eye, you are trapped within another animal's stomach where you will die slowly and painfully. Like it's nature is horrifying both on like a micro and a macro scale.
The ocean is terrible. It is terrible in the classic sense of terrible.
Like the thought of like being a nerve cell, even though they're not conscious, and just not being able to communicate when that is your job and you don't know why, like nightmare fuel.
Yeah, and I feel like I'm wondering how common that is for biologists, like not even just molecular or marine, but like all biologists. I wonder if there's a higher baseline level of empathy for the life that we study. There's a good question about TTX and the blood-brain barrier. So the fact that your body systems shut down before your consciousness shuts down, it could be blood-brain barrier related, right? Maybe TTX can't cross that blood-brain barrier. It could also be metabolism related, just like trafficking. Like TTX just doesn't traffic to the brain. I don't know the answer. It's a good question. I just don't know what the answer would be there. So super cool. So TTX's ability to shut down neurons makes it an excellent research tool. So this is what I was using TTX for. So the context that I was using TTX was not having to do anything with marine biology. I was using the neurotoxin for its property to shut neurons up, because what I was studying was their baseline synaptic strength. So neurons will talk to each other, and that strength is often polluted with the constant action potentials. Like action potentials are just like screaming, they're just like screaming, and then that causes the next neuron to scream. I wanted to study their baseline, like regular group chat that they have, where it's just like casual, whereas an action potential is much more chaotic. So to shut the neurons up, I added tetrodotoxin, and then I could hear the signal that they were sending each other when they're not screaming at each other, that was revealed when it's not being obfuscated by the screaming of action potentials.
Oh, interesting.
This is probably a video topic on its own. Of course, we were talking before stream how like if you want to make a science person really happy, just ask them what they studied.
Yep.
So I don't want to take up too much time with what I studied in graduate school, but it's a little teaser for when I eventually make this video. So there are a lot of mysteries regarding TTX, which again is really surprising to me because we've known about this molecule for decades, right? This is an old, quote unquote old, relatively like well characterized neurotoxin for its effects on biology, but we actually don't know a lot about this neurotoxin as a whole. And I think the first mystery that I want to present to chat, so we finally got to the part of the stream where I'm going to invite some chat interaction, is I want to just ask you, what do you call this thing? What is this thing? Is it a protein? What would you label this as? And I think that's a really interesting question to ask when we are talking about biology. Obviously it's a toxin, it's a neurotoxin, but like this is a question for my chemists in chat. Like, what do you call this thing? No, don't go looking up its like IUPAC name or whatever, the official chemistry name, but like what is this thing classified as?
Actually, I never thought about that before. I always thought that like just toxin was like like an acceptable term, and that was enough.
Yeah, so a toxin is not inherently a chemical feature, right? A toxin is something that exists because it reacts to biologics, but like this has a classification. It's going to make you mad when I reveal what that classification is, but there are some interesting answers in chat. So there's an observation out there from, I'm gonna butcher your name, I'm so sorry, Panucolo Klakos, saying that they can't see a carboxyl group, so a carbon, oxygen, oxygen, hydrogen group, so it's not a protein. Yeah, so if I ask you, is this a protein? This is a molecule that a biological organism makes, but is it a protein? It's not a protein. You can't see the signature of it being a protein. Nicholas says molecule, definitely a molecule. Seed says also, is it made of amino acids? So this is a question for my biologist in chat. Do you see amino acids here? No, it's very, very interesting. So a protein is a chain of amino acids, and if I pull up the amino acid chart, maybe this will help. So for those of you who don't know what an amino acid looks like, I have the chart here, and I want you to just look at the structure. And I'm not really a chemist myself, but I think that this is an interesting deepening of all of our understanding of chemistry, which is important. Again, like when I talk to you about tools, right? Like chemistry is still a tool that I have. I'm just really bad at using that tool.
Yeah, see, this is why I took the minimum amount of molecular and chemistry classes that I needed, because all these look the same to me. I can't parse them.
Yeah. Woody MI says, I see a carbon atom connected to 3 oxygens, so that's mildly cursed. It is. A TTX has a cursed structure. Like, I want to just point it out. Like, if you are a biologist in my chat, you're probably very familiar with what a protein looks like. It's a chain of these guys over here, right? They just link to one another, and it's pretty straightforward. And I want to, just show you this molecule, and look at this creepy cage. This is a cage molecule, right? This has a complex 3-dimensional structure that is not anything like a protein. And I think that is really, really neat. And so I just wanted to point that out, that this is not a protein, but a biological entity made this. And how could that be? That's kind of an interesting idea in of itself, right? Like the idea that we have the central dogma, right? Like, if you know anything about biology, it's that DNA makes RNA, makes protein. This is what everybody who takes a biology class learned. And this seems to be missing from this dogma, right?
Yeah.
And that's weird. Dreamless says it's a thing. It is a thing. It is a thing that a biological organism made, but it's not DNA, it's not RNA, it's not protein. So I actually saw the correct answer earlier in chat. What is this then? So this is what is called a small molecule. And I know you're going to hate me because I said the molecule thing. I know. It turns out molecule...
Scientists are terrible at naming things. Scientists are terrible at naming things. Sorry, I didn't mean to interrupt.
No. It's the indignation is very valuable because it doesn't have a fancy name, but it's not a protein. That's really what I wanted to point out. It's a molecule, right? It is something that an organism built, right? And so when you're thinking about the central dogma, information flows from DNA to RNA to protein, but you're actually neglecting a whole class of chemistry by ignoring this part of the central dogma, which is whatever organism, maybe it's Vibrio, the bacteria, maybe Vibrio made some TTX, but Vibrio is not coded the same way that proteins are. Thank you so much for the super chat, Cthulhu. Thank you again. Thank to the great old one. I think TTX is a very apt topic for a dark old one of a great old one to be watching. But thank you again. Thank
Absolutely.
you so much.
Wait, can I just point out a comment from Cthulhu Lulu because it made me laugh?
Yeah.
She called it a smolecule.
A smolecule. That's really good. I love that. So I just wanted to point this out because biological entities are more than just DNA, RNA, and protein. Your proteins also include enzymes, and those enzymes are capable of doing chemistry, and that chemistry is capable of making molecules with biological activity, right? So this is just for my biologists out there who have to remember that there is more to life than just proteins, DNA, and RNA. There's a bunch of other stuff that doesn't get talked about as much.
Can I ask something?
Yeah, go ahead.
Do you know why? Do you know why it's called a small molecule? Because, like, my very limited understanding of molecules is that that one's actually pretty big. And I don't know if you know this off the top of your head, especially because it's more of like a linguistic thing than it is a science thing.
Yeah, there's like a cutoff. There's like a protein.
Okay,
There's like a sorry.
there's like an arbitrary cutoff.
There's it's somewhat arbitrary, but this was more of the language adopted by a guy who was very influential in the pharmaceutical study. So there's a class of molecule called small molecule that fits under a certain molecular size that has good candidates of being biologically active, useful medicines. And we're actually going to talk a little bit about if TTX, has that kind of potential. Yeah, but it's small. Whether or not it is like therapeutically, it has therapeutic potential, that's remained to be seen. But it's a molecule and it's small, and maybe it has enough properties to be considered to be a useful therapeutic. We'll think about that in a later slide. But yeah, I just wanted to make it known that your body is doing chemistry, and to not neglect the chemistry that happens in your body. We're more than just biology.
I remember Hank Green once said that, like, people are like, there's all these chemicals in our food, and it's like everything is chemicals.
Yeah,
including us.
including us. Okay, so a lot of text on this slide, but that's because there's a big quote here. The pathway for TTX biomolecular synthesis is still a mystery. So by pathway, I mean the chemical pathway. What enzymes are doing what, in what order. Sorry to bring back the Orgo trauma for you, but like that's what I'm referring to by pathway, right? The organic chemistry steps that are required to start from a starting material to the ending material. This is actually not well known as far as I could tell. So as we've said multiple times on the stream so far, TTX is not synthesized by the pufferfish. It actually bioaccumulates in the pufferfish from bacterial origins. So I'm going to quote this. I think this is a textbook. I think this is in the citations in the video description. But I think it's important to read the words of the actual scientists who study this kind of thing. So although Tetrodotoxin has been the subject of focused biosynthetic inquiry for decades, again, that's what I said. Like, I was really surprised that we still don't know a lot about this molecule. Its assembly remains unclear, and no biosynthetic gene has yet been identified. This unsolved biosynthetic mystery has been exacerbated by a lack of reproducible organism and conditions wherein tetrodotoxin can be robustly produced in the laboratory. The diverse distribution amongst animals combined with this unusual structure, again that cursed cage, has suggested a bacterial origin coupled with either dietary acquisition or tetrodotoxin by animals from exogenous sources. Again, that fancy word just means from the outside, or endogenous acquisition via bacteriosymbiosis. So I think this is like, I love this aspect of science where, when we first brainstormed this idea, I was like, we have to know this answer, right? Like, this is just, I can imagine a couple of experiments you do and, zip zap, we have the answer. But this is a lot more complex than it seems on the surface. And I think that's kind of beautiful.
Yeah.
And tempting too. Do we know at least the name of the bacteria? So I learned from Octopus Lady's video that the leading candidate for the TTX production for the pufferfish is Vibrio. So Vibrio is a very interesting bacteria that I will talk a lot more about when I talk about biofilms at some point. I love Vibrio. We had one of the world's leading experts on Vibrio at my graduate school, and I've
wow.
I'm very hyped to talk about Vibrio, and I had no idea that Vibrio were capable of making TTX. So you learn something new every day. But here's maybe a good transition or a good opportunity to showcase, I think, the try-hardness of molecular biology. So I learned from your video that Vibrio makes TTX and that Vibrio lives inside of pufferfish. But
yes, or at least that's the hypothesis. One of the hypotheses that we have. Sorry, didn't mean to interrupt.
Oh, no. So, like, yeah, exactly. I like how you worded that, right? That's the leading hypothesis. And like, I want to challenge chat right now. Like, try to see it from my perspective. We know that Vibrio can make TTX. We know that it lives inside of pufferfish. I think to most people that's a done deal, right? That seems like a pretty straightforward answer to me. It's got to be Vibrio then.
Yeah.
So chat, let's take a little break while I have a sip of my water and tell me why that's not good enough for me. That I think that is very enticing to believe that the source of TTX for pufferfish is Vibrio, but for a molecular biologist, that's actually not enough. So I'll take a little bit of time to just brainstorm why that's not quite enough proof for me. And I'm wondering, I don't know, I don't want to like put you totally on the spot too, because that's bad form. But if Octopus Lady, if you want to take a stab at it, you are free to as well.
Yeah. I mean, I won't be able to extrapolate on it too much, but I do know that just straight out the gate, like correlation is not causation, right? That's like one of the mantras of being a scientist. Just because like two things are in proximity to each other doesn't mean that they are at all related. Just because like, you know, bacteria is around that ostensibly makes tetrodotoxin by itself, it doesn't necessarily mean that that's like where the pufferfish is getting it from. You need to do experiments and better research than that. That's about all I got though. From a molecular level, I can't like elaborate any further.
No, I mean that's great, right? Like something that I say all the time on my streams is correlation is not causation, and I think the angle that you are approaching it from is maybe pufferfish are getting their TTX from somewhere else that is not Vibrio, which is, I think, one weakness of this line of thought, right? And I want to point this out because it's so tempting to believe that the culprit is Vibrio because the story makes sense. And when I say, like, I say in my streams all the time that when I read a paper on stream, we're often trying to break hypotheses, and if you don't try to break hypotheses, you're not doing science correctly, right? We have to break this line of thought. And so I see some good contributions from chat right now hypothesizing why this isn't good enough for me, and like you're all right to a certain degree, right? I think Octopus Lady's angle of like it might be from somewhere else, it might not necessarily be from Vibrio in the real context of an actual live pufferfish. Sure, Vibrio might have the potential to make TTX, and maybe it does make TTX. I do believe that we do know that Vibrio can't make TTX, though.
Yeah, I do feel we're pretty confident on that.
Yeah.
Probably double check that, though.
And I think we're also pretty sure that pufferfish can take up Vibrio from the environment. But you see, like, every step of the story makes perfect sense, and it's very tempting to conclude that it's Vibrio. But I want, like, my audience in these streams to appreciate the fact that it's not true unless we rule out all the alternate hypotheses. And a good alternate hypothesis here is that maybe it's some other source, as you said, and that while it is technically possible for Vibrio to do this, it might not do it in a live pufferfish. And then, so the next question that becomes, how do we test that? How do we test whether or not it really is Vibrio? And I'll take a pause there. And so I think this is a lot harder for you to contribute here, Octopus Lady. But if you want to take a stab at it, there is that option for you. If not, my chat will, I think, wreck their brains. How do we test whether or not it really, really, truly is Vibrio in the pufferfish, knowing all these correlates about TTX production?
Yeah. Let me sit and think on that for a second.
Yeah,
So
no pressure.
we're trying to figure out if the TTX comes from Vibrio that we find in pufferfish. That's the question?
Yeah,
right?
if I don't want to give the answer away because I was just about to like word it in a very specific way, but like how do we know that in the wild the pufferfish's TTX comes from Vibrio?
Yeah. Let me just
I see a lot of interesting answers from the chat, and I'll say this. Give me your like, if you're in chat, you think of it in two ways. You can either contribute an idea that you think is practical, and then you can also think of this question as giving me an experiment that you think is maybe not practical but ideal. And then we can start to talk about why this problem is so difficult.
The only thing I can think of is like if we could somehow stick a pufferfish in an environment where we know for sure there's no Vibrio, and then put it in an environment where it's also eating from that environment. I don't know. That's not solid enough either.
No. But that's exactly what I'm looking for, right? Even if it's not practical,
Yeah.
give me something that even like ideally, what would you want to do? Don't worry about practicality.
Yeah. That's probably what I would do as a is trying to figure out if there's a way that I could just clear all the Vibrio out of an area, probably in a tank in the lab, and then also put food in the tank that the pufferfish will just eat on its own and just monitor its tetrodotoxin levels. Because yeah, what can like Fi mentioned this earlier, but like you can take a toxic pufferfish, and if you just feed it non like tetrodotoxin free food long enough, it loses its toxicity. So I don't know, that's that something like that.
Yeah.
That's the best I got.
No, I mean, that's great. And actually, there's a lot of commenters who are talking about the same idea, right? What if we gave the pufferfish its exact diet, but only removed the Vibrio? And I think that's a beautiful experiment. I think that would be great. That this is a test of sufficiency and necessity, right? If you take away the Vibrio from its environment, do you still get TTX? Keeping everything else in the diet the same.
Yeah.
And now we actually get to see why studying this is so complicated, because how do you even do that, right? You were running into this problem when you were trying to come up with an idea, because you can't just like take a normal pufferfish diet and then only remove the Vibrio. You could do it in the reverse, where you can reconstruct the pufferfish's entire diet piece by piece and then just not add Vibrio. But think about how expensive that would be. Every normal thing that a pufferfish eats, all the normal bacteria, all the normal fungi in their environment, but then just don't add that Vibrio. It's impossible, right?
How? Yeah, like how would you even do that? That sounds insane.
Yeah, and so like one other option, this is as a molecular, it's so funny because, you know, we are our training, I guess, because you were like, we'll put them in a pufferfish in the environment, remove it from the environment. I was thinking, what if we killed the Vibrio inside the pufferfish and see if they still accumulate TTX? But that runs into the same problem. How do you only kill the Vibrio? Right.
Yeah. And also like that's one of the hypotheses about how like TTX ends up inside a pufferfish. The other one is that they have no bacteria in their system, but they eat animals that like accumulate TTX in their system. So I'm not sure which one's correct.
Yeah, and it's really hard to test because then, you know, then I started to get a real appreciation for how deep and complex marine biology is from a molecular perspective. Because like, you know, before I was just like, you study fish, right? But now I'm like, oh, this is like a really hard problem.
Yes, there are so many hard problems in marine biology, but we could save that for the end.
Yeah, and I see some other comments in chat that are trying to work their way around, and I love that. So this is a regular, if you're from Octopus Lady's audience, this is a regular feature of my stream where I try to put you in the shoes of a scientist, because everybody is capable of this level of thought. Even if you don't have our level of training in the specific problems, you, by virtue of you having a human brain with you know, human brain cognitive function, you can do the same thing. Not to the same degree, but you can have fun with the science in the same way that we do, hypothesizing and designing experiments and stuff like that. Create another strand of bacteria that outcompetes Vibrio inside a pufferfish but doesn't produce TTX. So I'll just take this one as an example of like, the more you do to a system, the less like you want to strike the perfect balance of being as realistic as possible with enough control to make your data solid. So the problem I see with that particular experiment is that you have now introduced another covariable that you have to account for, which is you introduced a new bacteria, and now we don't know how that interacts with the rest of the very complicated system that is the pufferfish's environment. So that's not, it's an interesting idea, but I don't think that will get us closer to the answer. But this is, again, you know, it's fun to take a crack at these hard problems, but it's also really hard. But I think the failing is fun too, right? Like I could not come with a good example. I thought I could. I was very cocky going into this, but as I thought more and more about it, I was like, oh, there's a reason why we don't have an answer yet, is because some of that stuff
marine
just
biology
isn't there.
I think, well, I don't think just marine biology, but I think just like as you called it, which made me laugh really hard, touch grass biology.
Yeah.
Like, I think a lot of people think it's a lot simpler than it actually is. But there's a problem with grass, is that like if it's acting as some sort of variable, how the heck do you get rid of grass? Like, like that's the constant thing with dealing with like real life.
Yeah.
Not to say that molecular biology isn't real life, but like it is very hard to set up the world to be like a perfect experiment.
And I think this kind of touches back on our preference for what we study, because I love that about molecular biology, the fact that it is somewhat artificial, because in an artificial system, you have so much control. And I feel like I mean, this is kind of what I was getting the vibe of like I don't want to assume too much about who you are, in terms of traits. Well, you were saying earlier on that you have a sense of intuition and vibe based, and like, it feels like I am the opposite, in which I am anxious about control all the time, and if I don't have the data in front of me, I kind of panic. And like molecular biology is nice because you can have so much control over the system, and you don't have to worry about grass and how complicated actual organisms are. I hope that wasn't assuming too much about you.
No, absolutely not. That I don't disagree with anything that you just said. And like, there's definitely something extremely appealing about that. But there's also, to a degree, a sense of sterility, almost literally, that comes with that. I like the chaos of the natural world. That's like one of my favorite parts of marine biology, I think.
I think that's really beautiful to have both of these perspectives, because we have a lot of students, at least I have a lot of students in my chat. And I think hearing passionate people talk about what they like about their own thing can help them identify where they would like to put their investments into. There was another great comment about phage therapy. So bacteriophage can be extremely targeted. That's a really good idea. The question then becomes, how do you treat the pretend ocean environment for the pufferfish with enough phage to kill all the Vibrio? That's problem one if you want to do it the most accurate way to the pufferfish's environment. If you choose to do phage therapy in the artificial system that I recommended, that runs into the same problem of you created a disruption in the microbiota of the pufferfish, and now that can get kind of funky. Although that is pretty good. It would the idea is really solid, actually. I like the idea of using phage therapy to knock out the Vibrio, although I think a lot of microbiologists would take issue with that because of its effect on the microbiome. I will just say, as a practical thing, I don't know if there's enough money to do that. to develop a phage therapy specific for this problem.
Agreed.
But I want to make it very clear, I really like that idea. I'm sad that I didn't come up with it, because that's really, really good and is almost exactly what I would have wanted to do. But hearing that idea, I'm like, okay, I like that idea so much that I'm thinking more about how that would actually happen. And I think we run into the issue of there not being enough funding for to maybe set up that particular experiment.
Yeah, that'd be super cool. That'd be really neat.
That would be neat. That's a very neat idea, I will say.
So this might just be me being not very well informed macrobiologist, but like, what's the problem with doing it in the lab? Because that's how I would do it. If I somehow had access to this bacteriophage that just targeted Vibrio, I'd be like, all right, let's get pufferfish in tanks in a lab.
Yeah, so the lab part actually excludes the hypothesis that they are bioaccumulating from higher organisms in the food chain. It is a very targeted experiment just to rule out the bacteria that they're eating Vibrio, basically. It's not bad. That's why I really like that experiment. It's just that, like, I don't know, I'm very hard on myself, and I'm treating your ideas as if they were my own, which is actually a compliment, right? Like I'm like, I mean, that might be one of the highest compliments I can give, is that I didn't treat your idea like a student idea, like a young student idea, where it's like, oh, well, it's good, but I actually made it my own and then tried to think about the pros and cons. And the pro is that you can rule out that one hypothesis. The con is that it might not be the best use of time, because it is less accurate to the environment. Like you could get a very compelling result, whether or not it is real. So I'm saying it's a very good idea. I don't know if it's the best idea, but it is very good. And I want to make that clear. It is very, very good. It's just not the best one.
Yeah, no, yeah, definitely.
Very, very good. 5, you've been outsmarted by a chat member who studied video games. I'm very happy about that, honestly. I get such a sense of satisfaction when chat knows more biology than I do when I have less biology background. Shows that there is a reason to do these streams, and that is the reason why. There's so many good
I think as
contributions.
Yeah, I think that's also something as like it's very easy for scientists to get caught up in our own heads and with each other, and that sometimes it's really helpful to like show your work to like a layman or someone who doesn't have much of a background in biology, because they can, even though they don't have that background, they can look at things very differently than you, and that can be extremely helpful.
Yeah, exactly. And it's wonderful. Again, it's just proof that you can do it. It's just, if you want to do it professionally, you need to go to school, but you can do the thing. You can do the thinking. Okay. So I want to address one more hypothesis just to underscore sort of the difficulties of working with this system, is that we don't know the pathway that Vibrio uses for TTX, so we can't actually just knock out the TTX gene in Vibrio because we don't know what gene that is. There's a lot that is still not known about TTX, which again contributes to the problem. But yeah, excellent discussion we've had here, Chad. I will move on though. But yeah, that was awesome. I love that. Okay, so actually we have a Vibrio slide right here. Okay, so we can go through this quickly because I guess it is a very natural breakpoint, but I guess I'll just see if there were any additional points that I wanted to make on this slide. So we know that TTX accumulation is like the result of the puffer's diet. We know that Vibrio is one of the microflora inside the puffer's gut. We know that Vibrio can make TTX. We even know that Vibrio isolated from the puffer's gut can make TTX, but it's not still enough beyond the reasonable doubt to prove that Vibrio is the real culprit. And it's for the reasons that we have already said. Even though this story is extremely tempting, in order to be 100%, okay, I shouldn't say 100%, there's no 100% in science. In order to be 99.9% sure that the culprit is Vibrio, we need to do some experiments that are almost impossible. And so I think that is okay, right? Like it's kind of sucks because it's like, okay, this is an unsolved question forever, but that doesn't mean that we give up on it, and it also doesn't mean that we won't have a pretty good idea of what the source will eventually be, right? What I want to break down in my streams is that if you want to do science, you have to be comfortable with uncertainty. And to me, like if I had to bet money, I actually bet that it is Vibrio. Like I just talked a bunch of game about like, oh, it's not 100% sure it's Vibrio. If you were to force me, okay, I put $50 down and come back to me in 25 years, I bet it's Vibrio. This is very good evidence. It's just not perfect, right?
Yes. What's that? Is it Occam's razor, or was it the simplest explanation is usually the correct one?
Yeah, that's Occam's razor.
I don't think that's Occam. Oh, it is Occam's razor.
Yeah.
Okay, yeah. Nailed it. Yeah, I totally agree with you. Like that's like the lack of how does the TTX get from the bacteria into their skin and into their gonads and their intestines and all that, like that pathway being missing does technically does technically mean that we don't know where the TTX comes from. But like yeah, I agree. I would bet money on this too. I'd bet big money on this. I'd bet way more than $50. Like
maybe when I make Mr. Beast money, I'll bet more than $50.
Yeah, fair.
But yeah, so if there's one thing that my audience that I try to nurture, this idea of like, only Sith steal in absolutes, right? Like in science, things aren't right or wrong. It's, would I bet money on this today? Yes, no, or would I not bet? That kind of thing. Quote unquote scientific truth is just a series of probabilities. And I think it's very probable that it is Vibrio, but I wanted to illustrate that like the ceiling for how certain we can be is very, very high. And I think that's very interesting. Okay. So that's not the only mystery surrounding TTX. Of course, we still have today's stream topic, which, almost 2 hours in, we still haven't actually gotten to it, but here we are. How does TTX, how do pufferfish resist TTX poisoning? So we talked about the mystery surrounding its biosynthesis, around its bioaccumulation in the pufferfish, but now I want to challenge chat to come up with some hypotheses as to why the pufferfish doesn't just die. Like, again, 21 micrograms, a barely visible amount of salt, can kill half of the human adults, males, that come into contact with it. And so it's a miracle of biology that this organism, or that several different complex animals have evolved. Because, like, TTX not being poisoned to bacteria, dude, I'm sleeping. Like, that's totally obvious to me. It doesn't really surprise me. But a complex organism like a fish, where there are so many working parts that can be destroyed, it is really surprising to me that they can resist TTX at all. So I wonder what chat thinks about what the underlying mechanism is.
Yeah,
So
and
we'll say.
I can throw my hat into this ring. The only thing I could think of is that just their sodium ion channels are shaped differently to the point where TTX can't plug it up like it can in other animals.
Yeah. I'm actually seeing a couple of different hypotheses, and we've actually already, just within seconds of me asking this question, we have both of the hypotheses that we will be exploring today, including the one that Octopus Lady is suggesting. So I'm not going to spoil what that second hypothesis is, but I saw both of them, and they are the first. So like you, Octopus Lady, my first thought was similar to you. Maybe pufferfish have a special sodium channel that doesn't fit TTX as well as everyone else's sodium channel. There's another second compelling hypothesis that I saw fly by in chat, which I didn't think of myself, but it is in retrospect very obvious, and we will get into that one after this one. So first we will explore this hypothesis of the special sodium channel. I forgot to take this screen out, so this was the spoiler screen because I didn't want this lady to but that doesn't have to be there. So hypothesis 1, pufferfish have a unique sodium channel that doesn't interact with TTX, or does not as tightly bind to TTX as a normal one does. So what is the evidence for this? So when we take a look at the protein sequence, the amino acid sequence, if we take the building blocks, the gene that makes the sodium channel in the pufferfish, we actually see that there are some interesting substitutions in the TTX resistant pufferfish. Now, I'm going to explain what we're seeing on the screen here. So let me go ahead and get my pen out. This here is a picture of the pufferfish's sodium channel. It looks identical to, the sodium channel I showed earlier on stream, but it is very slightly different. And it is different in these two regions called domain 1 and domain 2. Unfortunately, I couldn't really identify where these were on this actual protein, but I do know that these domains are somewhere in the middle around that sodium pore. And when we take a look at the actual sequence of amino acids, if this is your first time seeing molecular biology, your proteins are made of mostly 20 to 21 different amino acids, and each of those amino acids is represented by a single letter. So some pufferfish have a domain 1 that looks like this, where it is, I'm going to butcher this because I didn't remember. It's, I think, arginine, methionine, threonine, glutamine, aspartate, arginine, tryptophan, glutamate. Oh god, I'm so bad. And serine. Serine's easy. S for serine. But the idea here is that in the pufferfish, they have two major differences than the regular channel, and it's these red letters here. So for domain 1, the pufferfish in this position has an N, and in a different pufferfish, instead of that N, they have a C. And this is actually quite different than other organisms where that substitution is not really a thing. Same thing for domain 2 down here. Oh god, I hope chat isn't full of people telling me that I got my amino acids wrong, which I probably did. But
you were very brave to try to remember them, man. I wouldn't have said anything at all.
I'm okay with being wrong. I think it's kind of fun to challenge myself to remember. But so I wanted to ask chat, how would you test this? So we have a tempting hypothesis, right? We took the pufferfish, we sequenced their DNA, and we took a look at how they build their sodium channels. And if our hypothesis is that their sodium channel is different, and it's this different sodium channel that causes TTX to not be as effective, how would you actually test this? And again, it's okay if you're not a biologist, if you're not a molecular biologist, just tell me what you would do ideally. Don't worry about if it's practical or not. If what you are suggesting is impractical, I might tell you how to make it practical in a real situation. And I don't know if you want to take a shot at that too.
Let me just sit and think
Yeah.
for a second. See, like, yeah, so like my whole thing with molecular biology is I don't know what you guys are capable of. Right?
Oh, just
like,
let me know what you want to do, and I'll tell you if it's possible.
Like, so you said that like they have a C in domain, they have like a C and a D in domain 1 and 2 that other animals don't have.
Yep.
Am I understanding that correctly? So switch them out. Can you just take those amino acids out and put in the ones that normally, quote unquote, normally go there, like for a different animal?
Yeah, so your approach would be to take the pufferfish and see if we can make it more sensitive by giving it a different sodium channel. Is that correct?
Yes. Yeah.
Yeah.
Or just switch the amino, switch the amino acids out. I assume they're like Lego, so you could just pluck one out and stick another one in.
Yeah, you can. Well, it's
okay.
technically more complicated than that, but the idea is possible, right? We can definitely do that.
Okay.
The thing that I will say is that while it is technically possible to do this, from what I could tell from the pufferfish research, is that pufferfish are not genetically tractable or genetically modifiable. Not because of something inherent about the pufferfish. They're not that different than other animals, but they are not. They have not been worked on enough to make them genetically modifiable. So here's where.
Sorry. What does that? What do you mean? Like, what? Like, we just don't know enough about like their genome to be able to like pick it apart, like we would other animals, or?
Yeah. So in order for an organism to be worth pouring money into genetically modifying, it has to have certain traits. So either it has to have a great importance for human health, and then we pour $1 billion into the research. It's helpful if they have a short lifespan and breed a lot, which I don't think fish generally have that problem. They have to be easily houseable, right? Fish are pretty hard to house compared to mice, for example.
Fair. I was about to say, well, and I was like, no, you're right. Like.
Yeah, mice, you need a very small cage, and you change their litter and feed them.
Water and salt, and it's a whole thing.
So it's not so much that their genomes are special in a kind of way, although everybody's genome is special. it's more that I think that there has not been enough attention to the, like, I'm not saying it's impossible. I'm just saying that with the current tools that we have available right now, it would be kind of hard to do that.
Interesting. See, I assume that, like, I don't know, that if you can switch genes out, in an animal, for some reason I assume that you could do that with any animal, regardless of what kind it was, that you didn't need to do any like prep work. But it sounds like there is actually prep work that needs to be done. That's really, I did not know that. That's very interesting.
And this is such a good segue to talk about this funny story I heard from graduate school. But mice were the forerunner for mammalian species that we work with, right? Lab mouse, it's synonymous with laboratory work at this point for, molecular biology. There's an alternate universe out there where it's actually the squirrel. There was one guy pushing the squirrel really, really hard as being the mammal to study, which I think good, because mice are way easier to work with than I assume squirrels are.
Squirrels I think are a lot smarter too, so they'd probably be bigger troublemakers.
And
they're bigger
and
destructive. I also just I saw someone in the chat ask, like, it was, where was it? It was like, are there TTX susceptible pufferfish? Which I thought was an interesting question. And I don't know if all pufferfish are like immune to TTX, but we do know that we have taken pufferfish in the wild that don't they don't naturally produce or they don't naturally have TTX in the wild, and we've brought them into a lab and we fed them food with TTX, and they didn't die, and they actually became toxic. So I don't know if all pufferfish are like that, but at least some of them, some of the non-toxic ones are not susceptible to TTX.
So
Did I
that
say that right?
yeah, so that implies that they have some sort of evolutionary history with TTX, even though they
Yeah.
evolved to not really care about it, which is kind of
Yeah.
crazy. So cool. Okay, so if we
Oh,
can't,
I'm gonna step away for just a minute. I'll be right back.
yep, no worries. So I'll just ask chat this, and I actually saw this answer in chat before. So but like, if it's not practical to do that experiment where we swap out the pufferfishes sodium channel, what could we do instead? Because that approach I think is really, really excellent. It's just the pufferfish is not the animal we want to be thinking about. What animal should we be thinking about? I've been having a lot of fun on stream, by the way. I don't know if you could tell, but like, it's so cool to be talking about this. Let's see. Oh, the porcupine fish? I don't know. It's a good question. The worm. Yeah. I mean, worms would be better. Worms are definitely easier to manipulate than fish are, I would say. Yeast, that's also interesting. Although yeast, I don't know if they conduct using sodium channels. I don't know enough about yeast to say if that would work. But worms have neurons, so that would be okay to do. I don't know if the flatworm is genetically tractable, right? The specific worm that makes TTX or has TTX, I should say. I don't know if that particular worm would do it. But yeah, what else? Humans, definitely not. Genetically modify another animal to the same sodium channel as the puffers. Yeah, more juice. That's a great idea. In which case, what animal do you think we go with? Definitely not worms. Definitely not humans. I mean, worms are a good option, though. C. elegans, maybe. It's definitely not a bad idea. Daniel Rario, so the zebrafish is an excellent choice as well, because zebrafish are actually genetically tractable. So Daniel Rario would be a very good option. So I actually like Wit I Am's answer of I think Daniel Rario is the zebrafish, right? I think so. Oh gosh, I gotta look that up. We had a world expert in Daniel Rario too, so it'd be really bad if I got this wrong. Zebrafish, great.
I am terrible at remembering scientific names, so
Yeah.
you're not alone.
Yeah. Dream asks, why not worm? So I don't know if the TTX worm is genetically tractable. C. elegans would be a better choice, just because C. elegans is the GMO organism. C. elegans is incredibly genetically tractable. So that would be interesting. And they do have neurons, so that would enable us to test all sorts of fun things with them. These are all really, really good options, honestly. Mouse, I saw mouse as another one. So I'll say that those are all really good options. Fruit fly, excellent option. Fruit fly would definitely be one that I would consider. But I think for this particular hypothesis, we don't have to make an animal suffer. Those are all excellent ideas. But I actually think that instead of thinking about an animal, we can think about cells. Cells, you don't have to kill an animal for, which is super good, right? So this is why I'm so like pro animal, right? Like when we do research, we don't just blindly sacrifice an animal to test something. We first have to make sure we justify the usage of animals. And so even though I think all of your answers are scientifically appropriate, they are not necessarily immediately the thing to do in a laboratory, because we don't have enough justification to hurt an animal for this kind of research. So to do this in cells is probably the better option. And indeed, we do have an experiment that does that here. So what you are seeing here, I'm sorry if you're red-green colorblind, but molecular biologists love green and red as a combination, which is super annoying, but this is the best image I could get of these cells. So these are hamster embryonic kidney cells. So these are kidney cells that have been isolated at some point from an embryonic mouse. And so the idea of swapping sodium channels, excellent idea, but instead of swapping a normal sodium channel into the pufferfish, we can swap the pufferfish's sodium channel into the rat cell. So this is a bit confusing. To swap the rat cell that has been mutated with the pufferfish amino acid change into the kidney cell. into
Gotcha.
yeah. So this is okay. So I just want to get kind of read from chat. How do you like this, right? So we took the rat sodium channel because it's more similar to the hamster. And then we changed it to have those same substitutions that the pufferfish has. We took that sodium channel, we put it into these kidney cells, and then it became 2,000 times more resistant to TTX. 2,000 times more resistant. So how satisfied are you with this answer? Are you very satisfied that this is what makes pufferfish so resistant to TTX? And I'll give you some time to think about that. And obviously, if you have some thoughts, I'd love to hear those too.
Yeah. I mean, to me, that seems like pretty solid proof. Unless there is some reason to believe that somewhere in the process of mutating rat sodium channels and then putting them in the hamster embryonic kidneys would somehow mess things up so much that like it was that process that made it TTX resistant. Like if that then I really can't think of and I'm assuming we know that it's not, otherwise we probably wouldn't be doing these experiments.
Oh, but you shouldn't assume.
You shouldn't assume. Very true.
So you are giving the molecular biologist too much credit. Your skepticism is actually exactly what I wanted to touch upon, right?
Okay.
I'm not super satisfied with this as an answer. I think it's pretty good. It's pretty interesting. But as you said, maybe we just broke the sodium channel, right? What's
that's a much
missing?
simpler way to say it. Yeah.
If you did the mutations out of the context of the pufferfish, maybe you just break the whole thing.
Yeah, that's a good point. I didn't even think of that.
I mean, you did. You said it in your words. I just
Yeah. I'm not always greatest. I'm not the greatest at always, like, I'll say things and then people will be like, "Oh," and then they'll say something that's deep and insightful and I'm like, "Yeah, that I did not derive that conclusion myself. Thank you for pointing that out."
Yeah, but I did use your words.
Right.
So what if we just broke it? In the process of mutation, maybe we just broke it. So how would you actually test this in a more realistic situation? Because maybe, how do we test that this mutation didn't just break the sodium channel entirely? How do we test that, chat? How do we test that?
How would we test that?
Yeah, it's interesting, right? How do we make sure that this is still a functioning sodium channel?
How do we test that? Can we do the same thing, but like mutate the sodium channel in a different way, and then put it in the hamster embryonic kidney cells, and then expose it to TTX? Does that make sense?
I wouldn't go with a different mutation just because we're going to run into the same thing. But I'll take your words and then apply it more broadly in the sense where we can do this mutation in a different context. What if we took regular neurons, and I saw this answer in chat, so good job, chat. What if we took neurons and we just changed their existing sodium channels to this mutant sodium channel, and then see if that neuron can still do neuron things?
Yeah, why didn't they do that? Because why didn't the researchers who did this? Why didn't they do that? Because this seems like a lot of extra steps.
My guess, okay, so in their paper they did check to see that the sodium channel still works. Like it does in these cells, right? But they're not neurons ultimately, right? So they can still open, they can still close, they let sodium in, like that appears to be working fine. But what I wanted to get at is that doesn't mean that this is that they're, because like what they tested for was, okay, they can let sodium in, cool. But in a neuron, like, can they do an action potential is like the big question that I still have in the back of my mind. And so the reason why they didn't is, my guess is because as a chatter has already said, neurons are really hard to culture. Kidney cells, embryo, like generally tissue that you get from, embryonic culture, they love growing. They just divide and divide and divide because, growing organism, right? A neuron is post-mitotic. Once you plate that sucker on a plate, if it dies, it dies. And then, I've personally grown neurons. They're tremendously difficult. It sucks. I don't recommend it. So I'm not really mad that they didn't do this in neurons. I actually think that this is fine evidence, although this specter of like, how bad did you actually screw this protein up? I saw a chat ask a question from chat that says, "I'm starting to believe that molecular biologists are never satisfied." That's true to a certain extent, but like I think the culture of molecular biology is that every paper is just one piece of evidence, right? Like no individual paper is ever enough to convince anybody of anything. Even this paper, I think this is cool. I think this is interesting. I actually think that this is pretty convincing, but there's still room to be more sure, right? There's still more room to be like, okay, this is definitely it, because there are other explanations that we have to rule out before we can definitively say that this is the case.
Interesting.
Yeah. So I actually think this is pretty good, right? I think that the fact that the sodium channels still work, even though they're not in the right, they're not in the correct cell, the fact that they still work shows that the actual structure of that channel is mostly intact, even with those substitutions. Whether or not they can still fire. And then even if you do put them in a neuron and they work or they don't work, that's also difficult to interpret because unless you grow up the whole animal, you could do it in a mouse, and then if the animal is largely normal, then you can make an argument. But you always, it's by definition an artificial system, so you're always going to be wondering, doing this strategy, whether or not you really have the correct answer, which is fine, right? That's why multiple scientists work on the same thing. But it is a little bit of juicy nuance to be like, science is hard.
Yeah.
Science is hard, but it's interesting. It's like really fun to do it, but it is frustrating at times.
Yep.
Okay. So what would we need for complete proof? So here's the hypothesis. Pufferfish have amino acid substitutions in their sodium channels that cause them to be resistant to TTX. One example that chat and you had recommended was swap a TTX protein, a channel protein from one organism to another. What's another experiment that we could do to try to figure this thing out? So we can do this swap, but what else could we do?
I do know that I do. So like so I went through these slides before, so I kind of already know the answers, but I genuinely forgot the first hypothesis. So I was like genuinely trying to suss out like a good experiment for that. But this one I do remember the answer to this one, so I'm just not going to say anything.
Gotcha. Yeah, so two different, and I don't think that even these two are perfect, right? But if we have these two arms, I think we can make a very convincing argument for this being the case, that these amino acid substitutions are what's actually true. So do the opposite. Give a pufferfish cell normal channels. So this was actually the first thing that you had recommended, is swapping in a normal one into the pufferfish. So we kind of already addressed this one. So swap the pufferfish's sodium channel for a non-TTX resistant sodium channel, and if it still tolerates TTX, this doesn't have to be lethal. So as I said to you earlier, this is not exactly practical because the pufferfish are not known, they're not a known genetically engineerable organism, but in order for us to have like maybe 80% certainty, we do need both of these answers, right? Because you can kind of see the beautiful way in which these two experiments work together to build up to the same hypothesis. And so here I want to shout out for my mole bios in chat, molecular biologists in chat. If you choose to study this discipline, and I don't actually know how much Octopus Lady you encounter this paradigm, but there's this paradigm in mole bio called necessity and sufficiency. And whenever you are designing experiments, you want to make sure that you are both thinking about the idea of necessity and the feeling of sufficiency. So what are necessity and sufficiency? So before I move on, have you heard of these terms in a scientific context?
No, this is the first I've heard of this.
I'm actually not surprised, because these are the ravings of like control freaks, right? I think molecular biologists are like control freaks because we have so much power over our given science, which again makes our thing more sterile and more not as representative of reality. But it does allow us to learn very specific things about ways things are set up, and if you do it often enough, then you can start to build into reality. So what are these ideas? So necessity is a question that you ask when you see, okay, does an organism or a cell or whatever, does it need a certain component for a certain function? Sufficiency is the opposite, as Chad has pointed out. Is this genetic component enough to be able to enable a certain function on its own? And so if you have these two answers, you can conquer like 90% of molecular biology. It's just, it gets very difficult to design experiments that can do both. And so if we take a look at the two experiments that we have suggested here, right, this first example is a test of sufficiency. Is the pufferfish's sodium channel enough to give the hamster embryonic kidney cell the ability to resist TTX? Is it sufficient? Can we give it and does it change? Necessity is the opposite. If we take away the pufferfishes and swap it with a normal, a different fish, maybe a porcupine fish's, sodium channel, does it still resist TTX? And this doesn't have to be lethal, right? If we're talking about animal rights. What's that?
I mean, it should. The porcupine fish should be able to resist it because they can also have TTX.
God damn it. When will I stop catching these strays?
It's all right, man. Like I said, you're a molecular biologist. Like, you're not cool enough to understand that's like multicellular.
It is
creatures.
True. I get very nervous around animals. Yeah, so Labella says they're not the paradigm in ecology/evolution. So that's really interesting to me because evolution and ecology are much closer to molecular biology than marine biology is. And we're seeing a lot of people say that these paradigms in math. That's also really interesting. I've never encountered this idea in math before, but that's actually really cool that we have that connection. That's very neat.
Yeah.
But yeah, so these are the two arms of any good molecular biology question. Is it sufficient and is it necessary? And chat, you and the octopus lady have beautifully reconstructed what I would consider to be ideal experiments to tackle this particular hypothesis. So that's awesome. I love that. So we do know from that hamster embryonic kidney experiment to some degree that it appears that the sodium channels are sufficient for TTX resistant asterisk in embryonic kidney cells using a rat as the baseline, right? But we have no idea if the pufferfish needs special sodium channels to resist TTX. This is a tricky idea, right? We have to be very thorough if we want to be sure of our hypotheses. Now, again, if I were to bet, I would bet that their channel is resistant, right? I would bet more than the Vibrio thing, right? I would bet $100 on this because this to me is pretty good evidence, but it's not complete. It's pretty good, but it's not fully there yet.
Mm-hmm. I would bet money on this too. Yeah.
This to me is very convincing, but I just for my audience, I wouldn't bet my life on it, but I would bet some money. Okay, so I did some digging, and I think an excellent question in chat came up earlier about the meaningfulness of TTX resistant and how much is like 2,000 times resistant. So here's a paper that I pulled about how much TTX actually is in a pufferfish, and I couldn't really get a good sense for the average amount of TTX in a particular pufferfish. This is specifically for Takifugu, albo plume views. Gosh.
Good. I'm gonna try.
Yes.
Let me try it. Takifugu albo plumbeus. Albo plumbeus is how, but I'm just guessing.
That sounds like that Rick and Morty thing, the plumbus.
Yeah, right.
So this paper is measuring the amount of TTX in spawning pufferfish, and I don't really have a full context of what that means. I take it to mean that they're just like young pufferfish who hang out around where they were hatched. But do you know what that means, like spawning pufferfish?
No. Well, yeah, like when they say spawning pufferfish, I'm assuming what they mean is that they are in the process of reproducing with each other right now.
Oh, well, okay, so they're adult pufferfish that are mating.
Yes.
Oh, so they're not like babies.
No.
Oh, okay. Good to have you on.
That's my yeah, no problem. Anytime.
Awesome. So this paper, the N's are kind of low, like 11 males in this region of Japan, whatever, right? But let's just, as an eyeball back of the envelope, napkin calculation, try to see whether or not that 2,000 times resistance actually means anything. So I looked up the data. We have the total amount of the TTX in these specimens and the average amount, and there's even a breakdown, but I'm going to ignore the breakdown because I just kind of want a very rough calculation, right? And I'm going with this top species, this, oh wait, what is the difference between these two graphs? I'm now forgetting. Oh, body weight, yeah. The top one is amount of TTX and the bottom is body weight. So I did some math. So the spawning pufferfish, so they have an average body weight, the males do, 36 grams, and they quote unquote tolerated, right? They can probably tolerate a lot more, but in the wild, this is how much they had. So the amount of TTX that they tolerated per gram body weight, 15.9 micrograms per gram of body weight. And then when you scale that to a human, we have a very large difference, right? So if we're talking about whether or not 2,000 times is enough to explain the TTX resistance, well, we'll just see. So I'm just going to pause here I feel like I didn't explain the chart very well. So the rightmost graph is how much tetrodotoxin this animal can tolerate per gram of body weight. And you can see that the pufferfish is way more tolerant than the human, right? Way more.
That's crazy.
And so is this bottom number 2,000 times less than the first number? So what does the calculation say? So when we normalize it, we actually see that if we scale it up to a human, this predicts that 7 micrograms of TTX are tolerated per gram body weight. So if we, in this hypothetical situation, if we swapped out our tetrodotoxin, our sodium channels, with that artificial rat modified pufferfish hybrid, whatever you want to call it, we get up to 7 micrograms of TTX per body weight, per gram of body weight. So we're missing half of that resistance. And this is, again, rough back of the envelope calculation. Some pufferfish have more, some pufferfish have less. But I'll say that this is an underestimation, right? Because pufferfish, they could probably take on a lot more TTX and be totally fine. This amount that I have listed here kills half of the people who are exposed to it, right? So it's a totally different ball game. Like, even though the difference is just 2-fold, in reality, it's probably like 200-fold. I just didn't have a way to calculate that. Yeah. It's shocking when you have the number, right?
Yeah, that's wild. Like, just pufferfish are so, I don't know. I've worked with pufferfish. I've worked with a lot of pufferfish in my life, and I love them. But they're like the goofiest fish. They're just goofballs. Like, it's just wild that they have like this, like,
superpower.
I don't know. Yeah. Like, they have a superpower. It's that. Yeah, they're the sweetest. They're the sweetest, silliest fish. And anyway, yeah, this is just leaving me a little speechless.
They're literally a goofball.
Yes.
So Mr. Winter asks a really good question. Does there have to be a single reason why pufferfish are resistant? And the answer is no. And that is an excellent transition to the next set of slides, where we will tackle the other hypothesis that chat mentioned. So hypothesis 2, maybe it's in addition, or maybe instead of the sodium channel, we don't know 100%, but maybe in addition to hypothesis 1, these are not mutually exclusive, maybe pufferfish have a unique way of soaking up and disposing of TTX, right? So maybe, the sodium channel thing doesn't completely explain the whole story. Maybe they have some other way that they can metabolize, block, soak up, whatever. So that's a possibility as well. So in order for this to be the case, the pufferfish has to make some TTX binding protein. And so this is a hunt, right? This is something that scientists have to look for. And I want to pose this question. This is mostly for my audience, but if your audience or you want to take a stab at it, that's great too. But I will preface this by saying that this is a hard question to answer, is how would you try and find this mysterious protein? And if you've been a longtime viewer of my channel, this might come easier to you than it would be otherwise. But, if you're not as familiar with molecular biology, again, don't worry about whether or not a certain experiment is possible. Just tell me what you would like to do, and I will try to convert it into an idea that can work. So how would you find, you have a pufferfish, you suspect that they make a protein that binds to TTX and maybe prevents it from destroying the pufferfish. How would you find that? while I have a sip of water. And again, this is hard. This is hard. Heraday says knockout testing one by one would work. That is technically possible. As I said, that it is not practically possible because you cannot. Well, there is. It's really hard to genetically modify a pufferfish. So I think that is a good answer. However, that cannot be converted into action reasonably.
What's knockout testing?
Excellent question.
Sorry,
So,
I didn't mean to interrupt your drinking.
no, all good. So knockout basically means we take the organism and we break their genes one by one until we get the result that we want. So we can go through the pufferfish's genome and we can say, hey, that looks like it could be a protein. And then hypothetically, we could break that gene and essentially break that protein. Again, this is an approach that could theoretically work. However, in the practical sense, not only are pufferfish very hard to genetically manipulate because no one has really done it, no one's done it like very seriously before, but the other reason why that approach is maybe suboptimal is because actually that's not really true. Let me take that back. I think most of why actually that does work because the genome is very long, right? What protein do you knock out? You just go one by one until you exhaust all, whatever 50,000 genes the pufferfish has. That's really a lot of work. So while it is technically going to get you the answer, it is not practically going to get you the answer, I think.
Curious, because we know a lot about zebrafish genome, right?
Yep.
So if we kind of line them up, like if we took a pufferfish genome and like I don't know, lined it up or put it right underneath an image of the zebrafish genome and there are genes that match like in the same spots, can we assume that the genes in the pufferfish do the same thing that the genes in the zebrafish do? Does that make sense?
You can assume, you have to test afterwards.
Okay.
But I like this line. We are sticklers. I like this line of thought though, so I'm curious as to where your head is heading. But I like where your brain's at.
Yeah, like that's the best I got. It's just knockout testing. But what I was thinking, and I don't even know if this is possible, was just like you try to extract and isolate every possible protein you could find in a pufferfish. And then I don't know, like separate each one into its own little, I don't know, test tube and throw Tetrodotoxin in there. And then see if it sucks up the Tetrodotoxin. I know that's that's impossible. That sounds like an impossible thing, but that was what I came up with.
Here's the thing. We can do that.
We can?
We can do that exact. That's actually so surprising. That
Wait, for real?
It's almost as if you had read the slides that were coming up, and the chat also has some of those similar answers. Actually, chat's answer, this paper that did this is actually from the 90s, and so chat, there are some answers from chat that are more modern. That also work, but the approach that you described just now is basically exactly what this paper does from the 90s.
Okay, I swear I did look at these slides in advance, but I swear I didn't remember that. I swear I used logic to get there.
No, I believe you because I added these slides in very late, so I don't think you would have read these.
Okay.
And the way that you kind of I think even if you read my slides, I don't think that you would have been able to not to throw shade on you, but I think the slides they need accompaniment. I don't think you would have gotten it just by looking at the slides.
Yeah, that is true. I remember looking at the slides that I did see. I was like, I don't know what he's trying to say on this slide. So because he's not saying anything, I'm just looking at it. So yeah.
I cannot describe to you how much fun I'm having right now, and the fact that because this is such the perfect illustration of why I love molecular biology so much is that it sounds intimidating, but it's actually not that bad, right? Like you pretty much if you don't get bogged down by what is possible and you just let your imagination go. Just talk to somebody who has experience and they can help you figure out what to do. So if you're thinking about molecular biology as a graduate study and you're afraid of it, I would say, there are good reasons not to study molecular biology. You don't like it, you don't want to do the work, that's all fine. Mm-hmm. If you think it's too hard, it could be. If you don't find that, I'm not saying that it's easy, but what I'm saying is that the nuts and bolts of what you need for molecular biology, I think a lot of people have that in them, just for the design aspect, right? Because that is exactly what they do. Like, I don't think that's possible. It is very possible, and we're gonna get into the clever way that they do that.
I'm proud of myself right now.
That's awesome. I don't think I would have been able to. Because I know the answer already. Like, I wouldn't, because when I read this paper, I was like, what did they do? Why did they do that? We have better technology these days. But it's fascinating from someone who doesn't have that background for you to intuit their answer is just so much fun. All right, so this is what chat was recommending, this idea of co-immunoprecipitation, although chat didn't say these words. Some people were recommending antibodies, like say you have an antibody against TTX, so an antibody that binds TTX, you bind that TTX and you use it like a fishing rod to fish out whatever is inside the pufferfish, right? So here I have a very crude drawing of a bead, like a tiny microscopic silica bead, and chemically you attach TTX to it. You just use chemistry and you just attach it. And then what you can do, and this is, again, animal research, we only do it when you really, really have to, and for very, very good reasons. You can grind up a pufferfish, and I know it's I mean, we saw a pufferfish pop earlier in the slide, right? Nature is metal, right? Like that happens in the wild. It's horrible. But, of course, the pufferfish are anesthetized and killed in a humane way. But you can then take their body, grind it up so that you're not missing any proteins. And then in that like pufferfish smoothie, you can run that across the beads and see what the TTX fishes out.
Oh my God, that's so smart. That's so clever. It's like a magnet.
Yeah, it's exactly like a magnet. Like it
oh my gosh,
is a very like, and when you hear it, it's like, oh, it's intuitive. Like that because molecular, but this science it has intuitive moments. You just have to work through memorizing everything about the mitochondria first, and then you can do the fun parts.
Yeah.
And so it is amazingly a simple process, and it is the more intuitive for a modern biologist, molecular biologist to think of, because these tools are like second nature to us at this point. But we didn't always have technology that could do this reliably, and so your recommendation of let's isolate every pufferfish protein and then just see what binds is what this group did. Okay, so what do they do instead? So they took the pufferfish. It starts the same way. You take that pufferfish, you homogenize it, is the fancy, not horrible way to say blenderize the pufferfish, right? And then what you can do is you can take the pufferfish juice and you can put it in a tube, and you can use what is called an ultracentrifuge. This is a machine that rotates your sample at, I think, above a million G or whatever, something ridiculous like that. So spins it around at faster speeds that you can possibly comprehend. And what that allows you to do is it allows you to do a process called fractionation, where you can separate the proteins by sizes, right? So when you say, can we isolate all the individual proteins of a pufferfish? The answer is kind of. We can separate them by size in a very crude way. Like, this is not precise, but you can see that, like, in this, drawing of a tube, that the small proteins that are not very dense, they float to the top. We have medium proteins, and then we got the big proteins on the bottom, right? So there's a spatial difference where we can kind of filter them by size, right? So we can separate them into fractions. Okay. So we can check each of these fractions for TTX binding activity. So we take this pufferfish juice that has been spun down, and we can take a pipette, and we can take the top 10 milliliters, the next 10 milliliters, et cetera, et cetera. We can take them fraction by fraction, and then we can take those proteins, and then we just see if they bind TTX. We just see if they manage to, and we have technology for that I don't want to get into, but essentially you can add the TTX to that fraction and then see if you can actually get binding to happen. You can measure something the thing is, these are chemistry methods and not really biology methods, so I don't want to get any of them incredibly incorrect, but you can shoot them into a fancy machine that gives you a signature, and you can tell whether or not tetrodotoxin is present or not present. But essentially, like, you can separate them by size, and then, you can kind of see where this is going, right? Like, okay, the third fraction of the 10 milliliters, that's the fraction that contains the TTX binding molecule. And then you take that fraction, you spin it down again, separate that even finer. and then you test it again, and you just keep on doing that until you can't do it anymore. And then you have one fraction that contains a protein of a specific size that you can then look into the genome and go, okay, I know this protein is 76 amino acids long. Let me look in the pufferfish genome and see if we can find a gene that is about 76 amino acids long.
Oh, wow, that's so smart.
Yeah. Oh, actually, that's the way we do it now. I wrote this on my slide. The way that they had to do it was they took that fraction, they used enzymes to chew it up, and then they sent those pieces, those jigsaw puzzles, into a machine that told them what the jigsaw puzzles were, and then they could reassemble that jigsaw puzzle and find out what that protein was. So it's the same idea, but a little different. Just wanted to make sure that I got it right.
That's still really impressive.
Yeah.
That's still crazy impressive.
Yeah, and a lot of classic molecular biology is this intuitive. You don't have to be like a god gamer programmer to like do biology.
Yeah.
Like it's you can do it. Mordru says it's almost like binary search but chemistry. Exactly, like this binary splitting that's an excellent way to put it. Is that you are splitting the fraction every time and isolating what you need to find. Very, very cool. Okay, so all of that preparation. Now, what did these scientists find? We have the Tetrodotoxin binding protein. So here it is. This is, they called it pufferfish cytoxin and tetrodotoxin binding protein, PSTB. So it's not an incredibly original name, but it is very descriptive, right? So in that process of filtering and testing to see if that binds and chewing it up and reassembling and all these little fancy tricks, they find this protein called PSTBP. And it's got this interesting pore structure, and I don't actually know where TTX would bind to this protein, but it's got this like 2 rings kind of structure, I think. Which I think is interesting, and I don't really know what the implications of that is. And along the mystery of understanding TTX, we don't actually know if this is this protein's true form. This structure is from AlphaFold. So AlphaFold recently won the Nobel Prize for Chemistry. It is a AI program, machine learning thing that allows you to fold proteins just knowing their amino acids. So here I have a picture. This is what we think the protein looks like, but again, with molecular biologists being hard asses, we don't know that for a fact. But yeah, at the end of this search, again, using the approach that Chat and you had suggested, we actually found the protein that binds TTX in the pufferfish, which is fascinating. Fascinating.
So is it both? Is it both? Do they have both a binding protein and their sodium ion channels are different?
Yeah, so these are 2 different proteins. So not only are their sodium channels a little different, they're also making a separate protein that binds to tetrodotoxin and, as a bonus, I guess, cytoxin. Yeah,
cytoxin, I don't know a ton about it, but I do know that there is algae. There's like dinoflagellates that produce that. It's a neurotoxin too.
The ocean is brutal.
Yes.
It is.
That's why it's the best.
So it goes so hard, man. Okay, but as I'm sure you're probably aware, it's not good enough that this protein binds TTX. We have to actually test whether or not this thing gives the pufferfish some TTX resistance, right? So again, necessity and sufficiency. So I don't actually know if these binding proteins are sufficient or necessary for pufferfish resistance. I don't actually think anybody does. And this is the really exciting part, because this is one of those moments where I'm like, how do they not know this? Right? This seems like such an obvious next step, but like that just shows that this, in our brainstorming, we had so many ideas that we threw out, but this one to me, I think spoke to me in a very deep way, because it is animating. It's an animating problem because it feels so incomplete. So, I mean, there's a ton of questions that still need answers. And so who's going to solve them? I mean, it's not me. I don't do science no more.
Yeah, who is going to solve them? Chat?
Yeah.
I'm looking very pointedly at Chat with a raised eyebrow. Just, hint, hint, nudge, nudge, wink, wink. If there's any interested molecular biologist scientists out there.
Yeah, or marine biologists that want to dip into molecular biology, right? Like, it's a problem from all angles.
Yeah.
Um,
Yeah, that's really cool.
do I have more slides? Okay, yeah, okay. This is the last one, and then we'll have some casual chat afterwards. But like, I have to end the stream like this every time because as a molecular biologist, it is my main motivation to always have like a health angle, right? Because that's how you get funding. little secret about molecular
Yeah.
biology is that you have to tie whatever you do into health in some way. Health or climate change are like too big, or like environmental health, right? Like there has to be some kind of health impact at the end and beginning of every grant. And so I just want to shout out that TTX is an interesting molecule outside of the context of the ocean, right? Like it has a lot of promise as an anti-cancer drug, as an anti-pain drug, and weirdly as a treatment for insomnia. The more we learn about TTX, the better its therapeutic potential will become. So anything that we can learn about how biologics interact with TTX is also going to be really, really useful, right? So, but not only did I read that TTX is this like interesting pharmaceutical option, I also read, and this is very brief and out of my field, but I also read that TTX may bioaccumulate in the ocean species that are important for human commerce and health as well, right? Like, I don't know, Octopus Lady, if you are, who's that Japanese fisherman? Masaru-kun or Masaru-san. He's a Japanese YouTuber who is a diving fisherman.
Oh, I'm not familiar with him. No.
So he basically shows off with the fact that he does free diving, and so he brings his camera with him, and you can see him like use his harpoon gun and watch him catch fish and fillet them and prepare them, and it's really, really interesting if you're into that kind of thing. But the thing that strikes me is how often he gives himself food poisoning.
Yeah, that'll do it, man. Like there's a lot of there's a lot of animals who eat other animals who eat other organisms that are toxic, and then it builds up in the animal's body, and then you eat it, and then you die or get very sick.
Exactly. Yeah. So there's this kind of concern that TTX might be, you know, due to climate change or warming oceans or whatever, that there might be an increased accumulation of TTX in species that will eventually make their way down into our food chain, right? So I'm a very human-centric creator because again, like I was raised in my training to always yell at agencies, hey, this is important for you, pointing at you directly right now in the fish that you eat, which is you know, it's pressing in that kind of way. And so I do think that like it is not just a for fun question like studies regarding TTX, but it actually does have like an actual impact on commerce, on health, et cetera, et cetera. So I think that's a neat way to kind of put a tie on the slide portion of this collab.
Yeah, definitely. Yeah, no. And I also just, you know, and I will say in defense of my beliefs is that I just also think it's important to answer this question because it's always good to expand human knowledge.
Yeah. I mean, I truly believe that too. And so I'm very glad that you say that because like my old institution used to be very like, we do basic research because basic research is important, but it's also because you don't know what you need to know later, right?
Yeah.
I feel like these days, I'm going to not say too much about it, but these days I feel the extra pressure to justify a capitalist reason why we need to do certain things. But that spirit is very much alive and well inside of me. So I'm glad that you pointed that out.
Yeah, it's just so much of like marine biology research is just like there really isn't any benefit to humanity for a lot of the stuff that we learn in that field. So it is really, I mean, don't get me wrong, there is a lot of stuff that will benefit humanity from research into marine biology, but there's also just a lot of like, we just wanted to know the answer to this, and someone gave us funding, and now we know the answer to this. It doesn't really benefit anyone, but you know. It was still important, regardless.
Yeah, and again, you don't know if we'll need that data later, right? That's always like
that's also true.
The appending on for like the crowd that thinks that every research dollar has to be efficient, right? That could be useful later.
So
that's what we'll end the slide portion. We'll take some questions and just chat with Chad and you know, hang out casually for a bit. I said at the beginning that I kind of wanted to get a read on whether or not you think that molecular biologists are if you get the control freak vibe from my presentation.
Oh my gosh, so it's kind of wild because marine biology, and I wonder to an extent if, like, I don't know, more like animal biology and zoology is similar to each other, because the level of like the amount of evidence that we need to definitively state something is true is way, 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 way less, like way less than what the standards that you guys hold yourself to. Um, like it's kind of blowing, like when you were talking about, like, yeah, the rat, the mutated rat, stuff in the hamster thing, and proved it to be like 2,000 times more resistant to tetrodotoxin. I'm like, yeah, that's good enough. And you were like, but it's not. And I'm like, oh, it would have been good enough in marine biology. Because, like, okay, like, I have so many stories. Like, I remember, this isn't necessarily marine biology, but I remember I wanted to figure out the uh, successful predation rate of lions. Like, what percent of the time do they successfully kill something to eat it, right? Um, and the number I kept seeing was 40%. They have a 40% success rate at killing things. And I was like, and I was about to put that in my script for the video I was making, but I was like, yeah, but where did that come from? And then I just kind of fell down this rabbit hole of, like, just people citing that number but not citing a source. I did eventually find the source, and it was this paper from, like, the early 1980s, where some researchers watched one pride of lions for one summer and just measured their predation rate, and they got 40%. And I'm like, that's not enough. Like, that's not, I think you called it an N sample? Is that what you called it?
Yeah. Number of N.
Yeah, like, number of N, like, that one pride of lions over one summer is not enough data to say that all lions have a 40%, like, successful predation rate. But every single paper that I, like, all the, when I was trying to find the source, I was reading all these other papers, stated it as if it's like a known and proven, like, well proven theory, or like fact, right? That, like, all lions, that's their predation rate. And it's like, that, no, it's one pride's predation rate.
Yeah,
That's not.
that pride could have been a skill issue.
Yeah, or maybe they're like the Golden State Warriors of, like, hunting prey, and they're, like, insanely great at it. Like, we don't know. Like, but everywhere you look, what's the predation rate of lions? 40%. Also, it's from a paper that's like 50 years old at this point, basically, almost. Like, but that's, like, the level of evidence. It's way lower, because I think this speaks to the type of world that, like, biologists like me, like, work in, is that, like, how are you going to get enough time and money and people to watch enough lions to
Yeah.
actually get a solid number on that?
I was gonna say,
Like,
we're actually very privileged because we can be control freaks, right? Like, I don't like,
yeah,
like, crapping on other disciplines is not like a thing that you should do, but you should acknowledge that, in, like, the reason why I don't do your science is because I am a control freak.
yeah.
But you can't be a control freak when you study something as complicated as the ocean, right? Like, it's just not,
Yeah.
it's not practical. You just can't, there's not enough money or resources to do that. So you have to be
Yeah.
accepting of a level of chaos. Yeah.
Yeah, or it's just like, genuinely, is there ever even enough money? Would there ever be enough money? Like, like, would there ever be enough resources? Because I remember I made a TikTok once about giant squid, and, like, I think I briefly mentioned something about the reproductive rates, or how giant squid reproduce, something about how giant squid reproduction, right? And we don't know anything about it. And someone on the TikTok left a comment being like, well, why don't we just, like, experiment with them in a lab? And it's like, what lab, man? Like, where in the world would we have a lab big enough to house not just one? Because if we're doing reproduction studies, we need 2 giant squid. Like, where? Like, the biggest fish tanks I know in the world are about 1,000,000 gallons, which I know that doesn't, I don't know if that means anything to anyone, but I'd say they're about, like, like, I've seen a 1,000,000 gallon tank. It's like 2.5 stories tall. I'm so bad with distance. Like, maybe, like, a couple 100 feet wide. You know, same thing with the length. Um, like, And I know giant squids that's not big enough. That tank is not big enough to house one giant squid, let alone two. And that's the biggest that we got in the world. Like, how would we ever be able to run experiments on giant squid? I don't know if it's possible. And like, yeah, there is an envy that I have towards your science, because there is so much more that you guys can answer than we can.
I think that that appreciation goes both ways in terms of like there's always this sense of insecurity that, and I actually got this comment on a video that I made yesterday, so it's a comment that did kind of stick out to me, talking about the fact that like, oh, you know, man is surprised the experimental system doesn't work after hacking off all of the confounding, like yeah, there's the sense of like we are control freaks, but like you have to think so carefully about how you control your experiment. Otherwise, your stuff is meaningless,
Yeah.
right? Like, there's a lot of battle for meaning in molecular biology. So, like, if there are interested students who like biology and like animals and like studying life, there's like two routes, right? Like, and like neither route is perfect, right? Because you say that there's envy that we have control, and I think that it is enviable. I like having control. That's why I gravitated toward this science, right? But there's also this envy of like, everything I do is in a dish, and I will never know if the thing that I studied actually mattered. And that's like
Fair.
a different kind of existential worry.
Fair. But like, I don't know, at the same time, if we somehow did manage to figure out how giant squids reproduce by sticking them in a tank big enough to house them, and even then, like, God, how would we even feed them? Like, I'm just sitting here trying to think about like the logistics of this.
Yeah,
Like, how would we even get them out of the deep without killing them? Like, there's so many insane steps that we'd have to do in
and
order to like.
they're really smart too, right? Like animal enrichment is not something that people really think about. But
Yeah.
like,
Well, like that's the other thing, is that we don't know how smart they are, because not to be mean to squid, but squid aren't as smart as their cuttlefish and octopus cousins.
oh, I didn't know
Um,
that.
yeah, at least I haven't seen a lot of information about squid intelligence. But like, yeah, they might be fine. That's another really good point. Is that like how, if they need enrichment, if they need stuff to keep them entertained, we don't even know. We wouldn't even, like, I don't think just throwing a ball in there is going to be enough to entertain them. Like, like, yeah, that there are just so many factors. It'd be insane. I would do it though if someone was just like, hey, if we have a giant squid and we need to figure out how to take care of it. I'm there front and center. This is going to be a nightmare, but like, to have the opportunity to do that, like, I would totally jump on it. But like, yeah, there is just like so much you have to do to like study real animals.
That is an interesting segue with something that I wanted to ask you. Is that like how much field work did you do? Like how much working with actual animals did you do?
So I didn't do a ton of field work, if I'm completely transparent, because I basically got my bachelor's degree in science, and then uh, never wanted to go to school again. So I didn't do a lot of research, but what I did do after I graduated from college is I went and actually worked in zoos and aquariums as what's called an aquarist, which is like a zookeeper, but we specifically take care of like aquatic animals. So I have a ton of experience working with animals, but just not in a research setting.
Gotcha. Because I was gonna draw this other distinction between like choosing what you want to choose if you're a biologist but don't know which route to go down. Is that like the touch grass biologists? They touch grass.
Yeah.
Is that if you're thinking that, oh, like the control freak thing sounds actually really appealing, you also have to keep in mind that you're inside all day unless you do some like field. There are a couple of molecular biologists who can do field work, but most of us are in the lab all day working with specimens that are too small for you to see with your human eye, right? Like if it is important to you that you experience biology in its context, then molecular biology will be quite difficult because you can't even see what you're working with. So
yeah, that's my issue with a lot of molecular or like microscopic stuff in general, is that like I need to be able to either see it with my own eyes or be able to easily imagine it, and I can't. My brain does not react to microscopic stuff the same. Like, it's like I don't know what's happening, I can't picture it, so I'm not interested. And I'm like, all right, brain, well, here's some cool brittle star pictures, and then my brain is happy again. But yeah, like, so let me ask you, like, what is it about molecular? Is it the control freakiness of molecular biology that you love, or like what is it about it that like got you into it?
Yeah, so similar how you have a story about when you were 16 and then like it just clicked for you. For
Mhm.
me, I was quite young as well. Although like back when I was an undergraduate, I considered doing psychology, like very seriously considered doing psychology as
Wow.
or cultural anthropology was another route that I wanted. I knew I wanted to do research of some
Ooh.
kind. But what settled on biology is there was a talk that a professor gave who I later did research for where he was talking about, okay, we studied this protein. It was the protein kinesin. So this is a protein that is in your cell that it's the protein that walks, right? It's the cargo delivery
Yeah.
man. Um,
Yeah.
yeah, so he was like, we're researchers who study this protein, and we want to know how this protein is involved with trafficking diseases in like Alzheimer's, for example. And I'm a young college student. I'm like, oh, this sounds really hard, sounds really complicated. And he was just like, yeah, so we just broke it. So we just broke it. We took this amino acid, we changed it to alanine, and then we just saw like, is it this? And then it wasn't that, so we just moved on. And then in the audience, I remember thinking, wait, I could do that. That's not that hard. That's like the same feeling that I want to give my audience is that coming up with
Yeah.
experiments, like coming up with the idea is not that hard. The hard part is execution, right? That's what you need the training, the wisdom,
Yeah.
but like just having an idea is actually pretty easy. Whether or not it's a good idea, that's
Yeah.
hard. But having an idea is not that hard. And it's the feeling of like, oh, there's something very like tactically satisfying about being able to work out this puzzle and like the battle between control. Someone in chat had asked if like talking about purity of subjects, which I'm guilty of adopting the language of like purity of subject, right? Like physics is more pure than chemistry, et cetera, et cetera. Even though that I agree with the
Yeah.
comment that it's judgmental inherently, but like
Yeah.
for me, molecular biology is great because there is some chaos, right? You are working with
Mhm.
life after all, but there's enough control that you can really express your creativity in a research setting. It is like for me the perfect balance of
Mm.
interesting chaos and the ability to wrangle that chaos. Whereas like my fellow V theory members Myelin and Sidere, they're both psychologists, and I think humans are like way too messy.
Yes. Yeah. um. Oh wait, I had another question. Wait, so like we kind of talked about this really briefly when we were first talking with each other about doing a collab. Is that like there aren't any mysteries left in molecular biology, um, which is like mind blowing to me because there are so many mysteries in there's so many
so
there's so
so
many
so
mysteries.
let me like qualify that.
Yeah, yeah, that's
Yeah.
what I was gonna ask. Like if you could like expand on that or explain it.
Yeah. So our working understanding of molecular biology, in my opinion, is pretty damn good. Like the process by which, for example, like so one of the hardest things to work out is the process by which an egg and a sperm become a huge like living thing, right? That's like
Mhm.
arguably one of the greatest puzzles in biology. And for an animal like C. elegans, that problem has been solved almost 100%. It's actually crazy for the worm. Like, we can know the
Okay.
destiny of every cell. As soon as that sperm comes in contact with the egg, we know pretty much every destiny of every cell that comes out. Like, you
Whoa.
watch that worm divide like under a microscope. It goes from one cell to two cells. We know exactly what cells that half will be. We know exactly what half that will be. They divide again. We still know those four pieces. We know this quadrant becomes these cells, this quadrant becomes these cells, et cetera, et cetera, et cetera. We have so much working knowledge about how biology happens, but of course there are still mysteries. I wanted to qualify it by saying that like the really really big stuff, I think,
Mhm.
and other biologists in chat, you can correct me if I'm wrong. Like, I think the frontier that we are pushing up against is like cancer biology and cognition, right? Brain stuff. Sure, there are other mysteries,
Mm.
right? But in terms of like, to me, the biggest problems, to my personal opinion, from my research background and my opinion, is cancer is still unsolved because we don't know what to do. I mean, we know a lot about it, but we don't know what to do about it. And then cognition, right? What combination of biology, chemistry, physics, quantum physics, what the heck is this thing we call the lived experience? Like, that's another unsolved thing. But like, you can understand why we haven't figured those out, right? Like, I think a lot of the other stuff, we don't have a full level of detail, but we have a pretty good approximation of what things are going on, to the point where like I think we had talked about this in stream before, where like the way to address this is like, how easy would it be for a senior in undergraduate to come up with a new idea to test in our respective disciplines? And for molecular biology, it's pretty hard. You have to do a lot of reading before you find a gap. It's not impossible, but for an undergraduate, you're 21 or 20, you just finished your last senior level course. It's still kind of hard for you to come up with a new idea and to come up with a good way to tackle the idea. But from what I'm getting from our conversation is that's probably a little easier in marine biology.
So it is and it isn't, because we hit that problem of like, there are so many unsolved mysteries, but the problem is that they're unsolved because like, how do you even test this stuff, right? Like another big unsolved mystery is we don't know why whales migrate. Like, and whales migrate really far. And humpback whales in particular, I think, are pretty interesting because they will travel like upwards of 5,000 miles somewhere and then just not eat for several months and then swim all the way back to where they started. And we have no idea why they do this. So like, sure, that's a question we can try to answer but like as a senior, undergraduate student, that's not going to happen, like, so it is interesting in the sense that like there are there are so many unsolved questions about so many different animals in the ocean, but like I don't know if we're ever going to be able to solve them. So it's there's a limited number of like questions that undergraduate students can ask themselves that they can reasonably solve by themselves. Does that make sense? So it's kind of similar to you, just different.
No, I get that. Because with the whales thing, right? I think the difference is that when I don't actually know if you had to do. Did you have to do a senior thesis where you had to come up with a new project?
Yeah, I did. It wasn't. I don't know. I mean, I don't know. I'm not super proud of it. It was just like how to do hermit crabs have a preference for what kind of shell they use?
But like so. I mean, that's kind of interesting. That's like not something that I ever really thought about. Like, what are the characteristics of like prime property?
Yeah,
what are
but like I don't know.
Looking at on Zillow.
me and my lab partner, I don't know. We weren't very good. This is another reason why I didn't really want to pursue higher education, is because I'm not the greatest at coming up with experiments. But like in order to get them to try to, in order to try and get them to pick a shell, right? We had to get them out of their original shell, and we didn't know how to do that. So, and like we found one paper that was just like, oh, if you put a flame, I feel so bad saying this. I still feel bad about this. If you put a flame like at the apex of their shell, they come running out. Like they start out. Yeah, they don't. They don't. They just stay in their shell and die. Like, and so.
You actually did this.
Yeah. Oh. We actually had to run the experiment. And so like, oh wait, did you guys not have to run the experiment?
No, because they're too expensive to do.
Oh.
They just had to come up with the idea. They just needed to do the idea, right? They just needed to prove that no one else had the answer. Like, it was an unsolved mystery, and that's why I was saying, like, it's hard to find a good question that someone didn't already answer unless you've already done a ton of reading. And just as a senior, like, you didn't do the reading, so it's really hard to come up with a new idea. Because when you start off the project, you go with the easy ones. How do we fight cancer? You do some Google scholaring, and then you go, okay, well, people have already done that. People have already done that. It gets hard to find your own little thing unless you do hours and hours and hours of research. But you actually had to do the thing. I mean, that's touch grass biology for you, right? Like, you were allowed to do that.
Yeah. I mean, if I think to your point earlier, I think if it was like, if we just had to come up with an idea and that was it, like, it'd be easy. Like, whatever. Let's try to figure out why whales migrate. Done. You know,
Yeah,
like I think
exactly,
that's why
exactly.
I think that's why they made us do the experiments, is because if it was just coming up with ideas, like what, 15 seconds, everyone's got one. You
Yeah,
know,
yeah, that's the
um,
difference that I wanted to highlight is that it would take your class 15
yeah,
seconds. For us, it would take hours.
yes, I get what you're saying now. Yes, absolutely, I totally agree with your assessment. Then,
Yeah, gotcha, gotcha. Oh, that's crazy that you were actually allowed to do lab work that was on under your
yeah,
own direction. Because
I
for us,
didn't like it.
it's always like cookie cutter stuff. Um,
Yeah, yeah. But yeah, I just that was that whole experiment was sort of a reason I was like, I don't think I'm cut out for grad school. I don't think I'm cut out to get like a PhD. I don't like running experiments.
yeah,
Um,
that is fair. That is fair.
um, although at the time I didn't realize that maybe I didn't even need to run experiments. I just need to go watch an animal once for like a summer and be like, there, that's enough data to prove
You will
to
remember.
back up my claim.
You will be remembered in history forever. You will
Yeah.
be cited so frequently.
For 50 years out, just like the lions.
Yeah. Yeah, I'm seeing that other people have similar experiences where it's graduation requirement. Yeah, yeah, we just had to come up with the idea. There's no practical way for us to buy all the lab supplies to do the science because it is expensive to do.
Yeah, that didn't even
Um,
occur to me. I always assumed that you like you I mean, you lab people. That sounds so derogatory, but I don't mean it like that.
if I can call you a touch grass biologist, you can call me a lab people. I think that's fine. That's fair. We can shake on that.
I always just assumed that you guys have everything that you need. Yeah, it didn't even occur to me that you would all need maybe different, I don't know, reagents and stuff in order to do your
Uh,
experiments.
I think if I told you how much it costs to operate a lab for a year, it would probably shock you.
Wait, how
It-
much does it cost you to
uh,
run a lab for a year?
for one
Wait, like wait, let me guess.
Okay,
Let me
okay,
guess.
okay.
Let me
Guess,
guess.
guess. For 1 year,
How,
a lab of about maybe 8 people, we'll say.
how many people?
8 people. It's just a
8 people?
maybe I'm pulling this number somewhat from my own ass, but like having seen
Okay.
a couple of lab budgets for the year, I feel like this is reasonable for one lab budget.
$500,000?
Oh, you're very close, like in excess of
Oh
a
yeah?
million. Like $1 million, million-ish, depending on some
Wow.
sciences more, some sciences less expensive. But yeah, to run that lab, and this is like the lab budget. It may or may not include people's salaries, is the thing, because sometimes they get their salaries from a different grant, training grant from the NIH or whatever. But
Oh.
like the operating cost of a molecular biology lab, that's smallish, medium-ish, it's about $1 million. It's yeah, so there's
Wow, okay, yeah.
no way we can
Yeah,
like have undergraduates come up with an idea and then test it. It would be very, very
yeah,
expensive.
yeah. Dang, that's still way more than I would have expected.
This is a very good. You were in the almost the correct order of magnitude, right? It's
Yeah,
practically
yeah.
the correct answer if I'm being honest.
Yeah, I feel like that's the case with a lot of science stuff, is like whatever number you think, just add a couple zeros, and that's how much it probably costs to do it.
Yeah, yeah.
Um,
Yeah. It's things cost a lot. Antibodies cost a lot.
yeah.
Oh, they call it modified eagle, but it should be money eagle. That's so funny. We have a juice for growing mammalian cells called modified eagle medium, but money eagle
Modified
medium is very.
eagle.
It's just
Oh,
the dude's name. The dude's name is Eagle.
oh, okay. I thought it had something to do with like bald eagles or something, and that would have been more interesting.
Always with the animals with you.
I know, right? Can't get them out of my head.
Okay. I had a lot of fun. This was an
Yeah, me
awesome,
too.
awesome collab. I
This was
think
great.
it went like, not that I thought it was gonna go poorly, but like I was really excited that you had stumbled upon. I shouldn't say stumbled upon. You intuited the experiment that they had run in the '90s almost to the exact detail, which
Yeah.
is just such a big molecular biology W, I think. Just like proof that if you will enable yourself to ideate freely, you can have good ideas, and this is a field where there are plenty of good ideas to be had. You just need help to get there. I hope you all learned a thing or two about TTX, kind of enjoyed the
Yeah.
mystery, the journey. I hope I hope you had a good time. I don't know what your
Yeah. No, I had a great time. This was really, really fun. We should do this again in the future.
can I be a little honest with the audience about the
Sure.
the energy pre-stream?
Oh yeah, go for it.
Octopus Lady was pretty nervous, pretty
Yeah.
nervous leading up to the stream. But I kept on telling her, like, if you just talk to me the way that we talk, like setting up the collab, dude, you're gonna be great. Like, I really felt like you were so natural on stream, and I think that the audience just had a blast. I mean, I had a blast. I think the audience had a blast too. So,
I hope
um,
so.
I think maybe what's limiting our collab is we gotta come up with another really good idea. Like this TTX, I cannot. We had like I think half a dozen ideas that we bounced back and forth, and just we couldn't nail one that was quite as resonant as this one. So I think whatever limits
Yeah.
our next collab is probably a good idea. So if you want to leave comments in the comment section of this live, we can I would love to do this
Yeah.
again. This has been really
Yeah.
fun.
Yeah, no, this is I really enjoyed myself. Yeah, I was extremely nervous. I'm very like yeah, I haven't done a live stream in a while. And I'm a very scripted person. So this is way more chill than I was expecting. So I had a great time.
So we'll go ahead and we'll end stream by doing whatever plugs we want to do. So I'll go first. So
Oh yeah.
I'm going to plug my long-term E. coli evolution experiment video. So if you haven't seen that, or if you're from Octopus Lady's audience and not familiar with my content, there was a guy who in the 80s thought, hey, what if we just grew bacteria forever and see how they evolved in the same unchanging environment for decades and decades and decades, and just watch evolution happen if they do evolve? How does it happen?
Yeah.
I think that's a really interesting experiment, and I really had a lot of fun making that video. So if you want to check that out, go ahead. It's in my videos tab. Do you have anything to plug?
Yeah, my channel here on YouTube at Octopus Lady, and then also as I mentioned before, check out specifically my video about pufferfish. It's linked in the description, and you can also follow me over on Blue Sky. If anyone here is on Blue Sky, although I'm not, to be completely transparent, the greatest at going on there, but I'm trying to get better.
Great. I will take that one last question that came in at the 10th hour, 11th hour. How does the body get rid of TTX? I don't really know. That's a question for a pharmacologist or a medicine person. But it is an interesting question. With that being said, I hope you all have a great rest of your day, and I will see you, I believe, next week for League of Legends.
League of Legends.
League of Legends, which I have I dread. So when I opened membership, I was like, I'll do gaming once a month, and if people want to play, they can play as like a way to interact with members who are so kind to support me month by month. But as the date approaches, I'm like bad at League of Legends, so the stress is increasing. But I think it'll be a lot of fun. So yeah, go check out Octopus Lady. Link in the description and in the pinned comments. I hope you have a great rest of your days. I will see you next week. Goodbye, y'all. Goodbye.
Bye. Bye, everyone.
Bye-bye.